All right, hey, we are live. All right, so tonight we're going back to like the old school kind of thing, man. We're going back to the old days of this channel where we were having discussions with young Earth creationists and we were talking about evolutionary theory. But we have a little bit different thing tonight. We actually got Williemaysfan. He has said we can call him Scott, who I've known from quite some time. He's been a young Earth creationist friend of mine for about 2 years now, I guess. And he is doing something that very few young Earth creationists I see ever do. He's doing the thing that what's his name? I'm trying to blank. Who I did a debate with, but Fillmore, Wayne Fillmore did. He's asking questions about evolution and actually wants to kind of ask to actual evolution people that actually know what they're talking about, rather than being filtered through a young Earth creationist mindset. So I've got the wonderful. Yeah, and I've got the wonderful Dapper Dino, wonderful Jackson Week, and the wonderful mustache Cheshire Vic, who's not an evolutionist. No, she is, but well, you're not like a biology person like they are.
I look like Beethoven to no relation.
No relation.
You've got a better mustache than he does. I've seen his mustache.
He's actually the one who got me these mustaches.
Yeah, he is.
You look like an Italian plumber. Yeah, that's true. All right, so let's get
Mario,
Lucci.
It's a me, Luigi.
It's a me.
So Willie, you gave us a whole bunch of stuff that I put in the video description. I know you kind of want to start on the Earth questions. So I think, how about you just throw them out there and we'll just kind of field them, and I'll let Dapper and Jackson take the evolution. We'll all kind of field the ones dealing with carbon dating that you're going to talk about. But go ahead and ask your questions, and I guarantee it, the people in here will give you the most up-to-date, honest answers that they are aware of.
Okay, great. So thank you for having me on. It's good to talk to you again and be on the show. For those that don't know me, I am a Christian. I am a young Earth creationist, deeply grounded in my faith. However, I'm in a place in my life where I'm pursuing truth. So if it leads me back to God, it's going to go back to God. If it leads me this way, I'm going to go that way, whatever that might be. So I'm not going to mention God, other than there's really two points of view here. You have a universe with God and one without. That's all I'm going to talk about as far as God. Everything else is going to be directed to you guys. So my first question is, it's about the Earth in general. Okay. I didn't research this. I'm just kind of going back on past knowledge, back in high school 30 years ago. It has to do with the crust of the Earth. It has to do with whether it's expanding or not. Am I right to say that the crust of the Earth is like 1 or 2 miles thick?
That is at least in the right order of magnitude, I believe. I am not quite good enough at geology to say for certain. It's also the case that the upper mantle sort of just transitions into crust material. It's not like there's a definite edge. It's sort of like going into space, right? Where does space start? You just kind of have to pick a convention, because as you go down, Earth radiates heat from the core, right? So as you go up from the core, it gradually gets cooler, and by the time it's cool enough to be solid, you've gone through that phase transition, gradually going up. But so to some extent, you can kind of say it's a little bit arbitrary where the crust stops when you're headed down. And I'm not sure what depth is usually given, although it is different. The crust is much thicker over continents, or at continents, than it is for oceanic plates. So there is that difference too, which is actually why they're oceanic plates, is because they're denser, heavier material. It ends up being thinner because it sinks lower.
So the short answer is that you're in the right ballpark, but it varies depending on where you look.
Yeah, and for space, there's the arbitrary Kármán line, right? That it actually is just an arbitrary line. We've said that at that point, anything above that is going to be considered to be space.
Yeah, it's roughly based on what most aircraft would need to go faster to maintain altitude than they could achieve via lift.
Here's the reason.
So go ahead. Sorry.
Here's the reason why I asked. I kind of set that up because I wanted to know, of all the fossils that we find, if the crust was a specific 1, 2, 3, 4 miles deep. Were all the fossils found within that area? And
Yes.
if not, have they been found lower than that?
No.
That's
So-
really what I wanted to get at.
So no humans have ever actually dug beneath the crust. So everything that we found in terms of fossils has come from actually not just the crust, but sort of the top part. Generally speaking, when fossils are found, it's not by like digging into a mine, although that does happen. Most fossils are simply found after the rocks that they're contained in have eroded on the surface, and then someone notices that there is a fossil there, and so then they start an excavation just around that site. There have been some fossils found in mines. Some coal mines in England have turned up some various fossils. There's some coal mines in the Appalachians that have turned up some fossils. Although in some cases they were not actually in their original geological environment. They were actually eroded out, then washed into a valley that then turned into a coal seam. But yeah, as a general rule, most fossils are found right at the surface.
I would
Okay.
also imagine you would run into a problem that as soon as you start getting too deep, they would start melting because it's all.
Interestingly enough, we do have some fossils that have been slightly metamorphosed and warped by the same processes that can bend and metamorphose rocks. So I recently read a paper describing an Ankylosaurus skull, and the skull was actually twisted slowly while it was underground because the rock it was contained in had been folded. So we get that.
All right, great. So I had other questions, but I'm just going to skip those. If we could jump to the Grand Canyon, that would be great. Not jump in, but jump to.
let's maybe not jump in that. You might die.
Yeah,
It's a deep hole in the ground.
It's very deep
All
and
right.
very steep.
They tell you gravity is a thing.
Yeah. So I don't really remember anything from school with the Grand Canyon, but now I know it's 6,000 feet deep. At the highest point, it's 12,000. I'm curious. First of all, Steve, what is a stratovolcano?
Okay, well, first of all, geology is not my thing. Now, I have looked at stratovolcanoes because I've done some stuff with Landon because he's a volcanologist. But I'm probably not going to be the best person to even try to attempt to answer that question, as opposed to like a cinder cone volcano and some other types of volcano. It's just not a field that I look into that much.
It's a minor question. It's not a major one.
But I will put out though that you would have to recognize that when Dapper was talking about these geological strata and stuff like that, most of them are very, very they're near the surface of the earth. You start going down and you start going back into the different eras. You're not going back down that deep as compared to what the crust is. I mean, it's about 20 some odd miles for continental. It's about 3 some odd kilometers for oceanic. But you're not like digging down to the mantle to find these fossils.
Okay, right. Okay, that's good. So, is it fair to say 100 feet?
You would have to look at there's a chart. There's called it's a relative and absolute dating system. You can look at the absolute dating system, see how it corresponds to the placement of what strata you would find a specific fossil.
Are you asking how deep we find fossils?
Yeah, he's asking for.
feet?
Yeah, exactly. Like let's say you're looking for like a
it-
500 million year old fossil, like a trilobite. What level do you know
okay.
and how deep you got to go?
I actually have my own answer because they find fossils in the Grand Canyon.
Yeah.
So I answered my own question. So the reason why I brought up Grand Canyon is because I found out that 32% of Earth's geological history is learned from the Grand Canyon. I did not know that.
I would say is present in the Grand
Right.
Canyon.
Yeah, it's there. Not that we learned specifically about it from the Grand Canyon alone.
Okay,
Right.
I wrote it wrong.
Yeah, but I mean
So-
that's a fairly minor point there.
Yeah.
Okay, so I think it's pretty substantial and why I should bring it up. So
And by the way, I've always said geology is my weak area. People, I've always said geology is my weakest area.
So I have a few questions about the Grand Canyon in reference to fossils. Let's see. Fossils, I mentioned the stratovolcano because it said when I was researching it, there are some stratovolcanoes in the Grand Canyon. One of them is as high as 12,000. And I think the Grand Canyon was like something like 7,000 above sea level or something, or I didn't write that down. I should have. But the reason why I brought it up was there are fossils and sea creatures, as well as fish teeth, approximately 7,000 to 8,000 feet above sea level in limestone
Mhm.
at the Grand Canyon. So my first question is, how is limestone found at 7,000 or 8,000 feet above sea level at the Grand Canyon? And then the second would be, how are they finding fossils of sea creatures and fish teeth?
I
Well, the
actually didn't know this
limestone
one.
was a well. That's uplift.
Uplift, yeah.
You have these layers forming. They're forming in a low area because limestone is formed from accretion of various phytoplankton, which die. Their shells precipitate down. They get compacted, concreted. That's limestone, or that's at least one way it forms. And so this gets over time. This gets uplifted by tectonic activity. So while it may be down here originally, it can get slowly over time raised and raised and raised, which is how you end up with like these gigantic coral formations in Texas, which are like mountains, but they're nowhere near, sea level.
So one thing I want to point out is, if you look at the period in which I would say mainstream geology is going to say that the Grand Canyon is forming in terms of the rocks, right? It goes all the way from very, very early on, but most of the fossil-bearing rocks that you're seeing are going to be from the Cambrian all the way towards the end of the Permian.
When was the Cambrian?
The Cambrian was about 600 million years ago. Is about when it starts.
540 to 480 million years ago.
Yeah, there you go. I was going one less decimal worth of precision.
Okay, go ahead.
Sorry.
But
It's a few million years here and there. Come on.
Yeah, so basically it's going from 540 million to about 270.
What'd you say the whole
Permian?
Paleozoic? Oh yeah, about 250 or so. Yeah,
250, 270. Yeah, okay. So during this period, towards the beginning of this, the continents are pushing together, and so North America is actually pushing up against the Eurasian plate, and this is when the Appalachians are forming. And what we see now with the Rockies and the Sierra Nevadas, and those essentially didn't exist. The whole area was flat and sloped mostly downwards as you went west. And so during periods of high temperature, when there are no polar ice caps, which is actually the sort of default on Earth, we are in an unusual time geologically speaking. So during that time, sea levels were high. And so continental crust that was relatively low elevation, which included at the time most of Western North America, tended to be either frequently flooded or it tended to have sea transgressions where the sea actually comes up onto the continental crust. Now, as Pangea starts to break up towards the end of the Permian, the direction that North America is traveling reverses. And so that's where we have right now, where North America is headed west and it's subducting the Pacific plates. The Pacific plate is sliding underneath the North American plate. And that's, as it goes down, it starts to melt. But because it's lower density than the mantle underneath it, that melted oceanic crust pushes up on the western edge of North America. So the western edge of North America is now much, much higher in elevation than it would have been, say, in the Cambrian or even the Permian. And so the reason that we're getting all of these marine strata is simply because they were pushed up by volcanic activity.
Yeah. So
now, this volcanic activity doesn't always result in an actual, like cinder cone or something.
Mm-hmm.
Sometimes it just results in the land getting taller and then the pressure can be relieved elsewhere in an actual, volcanic mountain that erupts. And so we have records of this volcanic activity all through the Mesozoic. It gets especially high during certain periods in the Jurassic and the Cretaceous. It's currently at sort of a lull, but it's still fairly active. You remember, I don't remember, but you were aware of Mount St. Helens and things like that.
Yeah,
so
I was going to bring that up.
Yeah, so this is all the result of sort of the same process of now that the North American plate is moving west, that west coast gets all the mountain building and volcanism.
Yeah, I mean, to really make it really simply, Scott, think about it this way. You asked why there are fish fossils. Why is there limestone? All that area once used to be underwater. It used to be an ocean. As you have volcanic activity and that what used to be the floor seabed got lifted up. That's called uplift. And as it got lifted up, the water got displaced. Right? So and eventually it got so high there's just no more ocean. The ocean's got completely getting displaced, but you end up still having the same fossils that were in the ocean bed. now
Right.
above sea level that you can go check out without having to go diving, basically.
And I also want to point out, since we're doing this sort of in the context of young Earth creationism, I know that generally speaking, I don't want to assume that this is your position, Scott, but generally speaking, most creationists would say that the reason that we have these marine fossils high up on these mountains is because the flood was covering them. But if you look at the fossils that we find in the Appalachian Mountains, which, remember, according to the sort of mainstream geology, would have been actually about as tall or taller than the Himalayas at the time where the continents start to break up. We don't find any marine fossils on those, even though right now they're much lower. So presumably, if this formed during a flood, we would find the same kind of marine fossils in the Appalachians. Instead, we find very terrestrial animals. We find things like dinosaurs. And yeah, in fact, we do find a lot of dinosaurs in the Appalachians. So the standard explanation from young Earth creationists kind of gets turned on its head once you look at other mountains other than, say, the Himalayas or the Rockies, which are both recent formations.
I would say even within the context of the ones that have fauna on them or fossil fauna on them, they're often deep sea fauna things. So these mountains would have had to have been under extremely deep water because they have things like crinoids.
Yeah, much more than 15
benthic organisms. Yeah, we're talking about, these things are way, way deep down. So the mountains would have to be under extremely deep water, which would mean the organisms on the ark would have to deal with oxygen constraints, which would be quite stressful. And it comes with lots of issues of its own. So
I don't want to get too much into the flood, but I think, does that sort of answer at least what the sort of the mainstream of science is
but it's
saying
good,
about these fossils?
It's definitely good to note, though. We're talking about what we would expect. Jester and I are a lot on Caffeine Corner. We say, hey, look, if this is the case, what are we going to expect, right? And if creation was true, we would expect a more homogeneous distribution of these types of fossils, and we don't see that. We see an actual stratigraphy of them. We see them going to be in layers, and we can predict and say, hey, look, based upon this type of fossil that we would expect, like a trilobite, okay, that goes back 540 some odd million years. What strata is it going to be in? What absolute layer is going to go in? Where can we go find this? And boom, they find it. There's a predictability there that does not exist in a creationist model.
correct. Which is why, if we are talking about, if we do mention dating methods later, that's why I want to bring up index fossils, but we haven't gotten there yet.
Absolutely. All right, go on, Scott.
Well, I did mention that because I was going to say Noah's flood, but you covered it. And that's fine.
Fair enough.
You brought up the Himalayas. I was going to bring up, 29,000 feet above sea level.
Absolutely.
How are their cephalopods, there? And
because they're underwater.
I'm not sure that we're actually finding many cephalopods, but actually there's a slightly different explanation there. So the Himalayan mountains are between the Indian plate and the Eurasian plate. And for most of basically the Phanerozoic, which is the period from the Cambrian all the way until today, the Indian subcontinent was moving sort of north and then sort of northeast and then sort of arced northwest. And it wasn't until fairly recently that the Indian plate actually collided with the Eurasian plate. And in this case, you get, instead of an oceanic plate and a continental plate meeting like we have in Western North America, we have two continental plates. Yeah. And when that happens, the underlying strata actually buckles. It's sort of like if you push two pieces of paper together, you might get them to push up in the middle.
Yeah, exactly. So it'd be a subduction where one goes under the other, they push together and push up together.
Right. Because they're both roughly the same density. So rather than one sinking under, they just buckle up.
Right.
And that's also how the Appalachians formed in the original Pangea, because that was the North American plate pushing against the western section of the Eurasian plate. And so
Eurasia.
Yeah, it was a similar process in that it's still a result of continental drift or plate tectonics, but in this case, it's not volcanic. It's simply of uplift through folding of the strata. So if you were to take a cross section of the Himalayas, you would actually see that the rock there is full of like sort of like W shapes, like it goes up and down because it's been folded.
Like a ripple.
Right. And so what that's doing at first, at the very beginning stages, which is going to end up being the highest parts, it's actually pushing up some of that ocean, some of that crust that's still continental but was underwater. And so that's why we're getting all these marine organisms pushed up to the top of Everest.
All right, let's jump to evolution.
Yay.
Absolutely.
Hey, something I know about. Yeah.
All right, so this first part is mainly for me, not for you. So evolution, science is a methodology. It observation, measuring, testing, that repeatable.
Mhm.
Here's the question: how does science use these methods to test evolution?
Jackson.
Well,
Do your thing,
wouldn't you
Jackson
like
Wheat.
to know? Okay. So there are, well, the very broad definition of evolution is simply a genetic change in a population over generations, descent with modification, allele frequency change over generations. It's some variant on changing the genetics of a population over time, right? Which also impacts their morphology, their body phenotype, whatever you want to call it. Well, in one sense, this is very trivial. You can very trivially change the genetics of a population by bacterial experiments are very popular in a sense because you can mutate them, or viral experiments, you can mutate them and see how they change. You can watch the fitness change based on certain mutations. The Richard Linsky E. coli experiment's one of the most famous examples, if not the most famous, because you had a population which duplicated a region of a gene and then moved it over to another gene, which caused this new phenotype. They can now do. What they basically did was they had two mutations, which allowed them to do a new thing. Right, the exact biochemistry of it isn't really relevant, but they can do this new thing that they couldn't do before based on two mutations that occurred in one lineage.
It's actually more they could do something, an old thing they could do in a different way.
Right.
Yeah.
So they don't have I believe the Linsky team removed their plastid, so they didn't have as much variability as these bacteria do in the wild because they have their plastids, which are changing constantly, things like that. But there are other experiments. So changing the genetic code is relatively trivial, but something that would be less trivial would be like, how could we observe new traits in multicellular organisms, or how could we observe, say, multicellularity arising in a population, right? How could we go from an organism which is made of a single cell to an organism which is made of multiple cells? And there are loads of experiments with this, experiments with selection based on gravity. You take this population of unicellular organisms, usually yeast, Saccharomyces cerevisiae, and you have them precipitate out. Do what?
My favorite fungus, it's brewer's yeast.
Yeah, you precipitate them out in like a liquid, and the ones who reach the bottom first, you take them and you put them in your next jar. You let them sit, let them settle out again. Take those, and you keep upping the time that it takes for them to settle out. Whoever can settle out the fastest, they are selected for. The ones who don't settle out the fastest are taken out of the gene pool, right? So over time, what you get when you run this sort of experiment is they start clumping together. And again, there are mutations that are involved in this, and researchers have figured out exactly which mutations are involved in that. But they start clumping together. The cells, divide, but instead of budding off from each other, they stick together. And so they're larger, they're heavier, and they can precipitate down faster. Or you can do this with a friend of mine who researches algae, Matthew Herron. He did this with predators. He took this unicellular algae. He put a whole bunch of them in this environment, and he released a predator which eats these a micro predator which eats these algae. And again, what it caused was they clumped together. The algae would clump together. At first, when they started clumping together, they were trying out what sequences would work. How many cells can clump together and still be functional? And so you get anywhere from 2 cells clumping together to 100 cells clumping together. But you have a selection pressure, right? The environment is selecting cells who are too large to be eaten by the predator, and yet are small enough such that they can still divide resources over their entire area, so that some of the cells aren't dying off and it slows them down or whatever, right? So that's another way that you are inducing multicellularity in this originally unicellular population. And there are loads and loads and loads of experiments like that. There's an experiment with fruit flies, Drosophila pseudobscura, in which these researchers raise them on raised two different populations on two different sugars. One was on maltose and one was starch. And after a while, they released the flies back together, but the flies couldn't reproduce with each other. They no longer saw flies of the opposite food source as mates.
That was Dodd's
And the
experiment, right?
Weren't they
what?
also
That was
chemically
Dodd's experiment.
Yes, that
Weren't
was
there also chemical barriers to reproduction as well as behavioral barriers in that one?
That was
There-
sexual
there-
selection.
Yes. Yeah, because the bacteria in their gut, Lactobacillus plantarum, releases metabolites, and we all have gut microbiota that releases metabolites, which regulates homeostasis. And what this did, what Lactobacillus did, was it gave off certain metabolites. So if you were the maltose flies, you're feeding it maltose, it's breaking the sugar down. It's telling you, or it's telling the flies, only reproduce with other flies that are breaking this down, breaking maltose down in their diet. And so you're right, there was a chemical barrier, this metabolites, that is influencing their behavior to not reproduce with the other one. So that's speciation, right? You got the populations, which are now, because they're not interbreeding, there's no gene flow between these populations. So they're going to go on separate evolutionary paths. They're going to undergo genetic drift and their own selective pressures, which is going to cause them to separate genetically. And so we can test basically all of the mechanisms of evolution: natural selection, sexual selection, speciation. We can test all of these things in the lab, and we have observations in the field for them. So yeah, there are loads and loads of experiments.
And I just put a link in the video description, or something in the live chat as well. What we would expect, Scott. Right now, there's an evolution going on in humans of this median artery. 35% of humans have it. They expect in the next 80 years that 100% of people have it. It is a new structure that has only been coming around as of lately, but eventually that will go through the entire population. So eventually all humans will have this new structure. This is something that we predict for evolutionary processes. We would predict new structurings being formed frequently and changing and evolving. That is how we get new structures from existing structures. So this is an evolution in action, is this new median artery that is right now in real time seeing people put forth this gene where people now have an extra artery they didn't have before.
So can I ask, Scott, do you find all those examples that I gave, do you find those to be compelling examples of evolution in the laboratory?
No.
Why?
Why not?
Well, I want to ask a really clear question first. Do you think that they meet the definition for evolution that is used by sort of the mainstream of biology?
Sure.
I think it's great that we can do these things with flies and and speciation and starch and sugar, and I think it's great that we can take different bacteria and make them grow and split off and grow and split off, and the others die. I get all that. Like, okay. I totally get all that. I guess it may have been. I don't know if it was too small of a question or too big of a question, because.
Go ahead.
I can't be anything but honest.
No, please.
Yeah,
that's what we want.
Bacteria and flies. I'm more interested in us. I'm more interested in animals. I'm more interested in.
So it happens, actually.
What happens?
Well, animals. Well, part of the problem is there are observable examples of things like speciation. We can observe natural selection, sexual selection, etc. in the environment, in nature. The problem is animals often have longer gestation times. They have longer generation times, and so it gets hard to measure some of these things. Like, if we want to know, how have elephants evolved, you have to look at the fossil record, because each one of us is only going to see a couple generations of elephants within our lifetime. So you have to look at. A
female elephant gives birth about every 2 years.
Exactly.
At maximum, it's almost impossible to watch real-time evolution and say,
although,
which is a bad term, but
which is why you have to go with things that are smaller. If you want an animal example, I can give you one. The marbled crawfish, Procambarus virginalis, had a whole genome duplication event. It duplicated the number of sets of chromosomes that it has. So it had 2 sets, diploid, that's what we have. We have 2 sets. But now it has 3. It's triploid, right? And because it has this new set, it makes it genetically incompatible with the parental species, which was Procambarus phallax, right? They can't mate anymore. Do you consider that evolution?
Going from 2 sets of chromosomes to 3.
Yeah, you have a whole genome duplication which separates them as a species. They can no longer reproduce.
I think where I and probably many other Christians haven't kind of ingrained in our mind is the whole, it goes from a pen to a pencil. I don't think we as a society, as a Christian society, boil it down or bottleneck it down to something like specific like that. It's whenever I'm in a discussion about that, it's always, how did a crocodile come from an alligator and come from a,
can I
gecko? You know what I mean? Yeah.
Yeah, I think what we've got here is maybe a misunderstanding of evolution in that it wouldn't be a pen to a pencil, it would be another kind of pencil or another kind of pen.
Yeah, right. I think one thing. Oh, sorry. I think one thing is analogies to man-made objects are always bad analogies in evolution. It's almost never a good idea to make them. Fundamentally, man-made tools. They don't reproduce. They're not.
I'll put it this way: a crocodile isn't going to become an alligator or vice versa.
Both of these came from an earlier crocodilomorph, a group that is ancestral to both. Sure, a crocodile-like ancestor. Both modern alligators and modern crocodiles came from a crocodile-like ancestor.
You would never see one turning into the other one, especially when it's still alive, or one giving birth to another one. I find that usually the best analogy for that is language. So at no point did a Latin-speaking parent give birth to a Spanish-speaking child.
Right.
Yeah.
It is just the language changed and morphed over time based on social pressures and where they lived, et cetera, et cetera.
And it changed differently in different places, which is also another really good analogy. Actually, languages are a great analogy to evolution because, with the exception of some like Creoles or Pidgins, which we'll just leave out for now, languages divide in basically the same way that organisms do. They divide into a nested hierarchy where all of the Romance languages. So let's say we take French Romanche, Spanish, Catalan. These are all West. They're not just Romance languages. They're West Romance languages. And Spanish and Portuguese are actually Iberian West Romance languages. And all the Romance languages grouped together with Greek and Etruscan as this. Well, actually, Greek. So Latin and Etruscan and the Romance languages all grouped together as the sort of Italic language family, and then that groups together with Greek as another family whose name I forget, and then that groups together into Western Proto-Indo-European languages, and then ultimately all of those, including all the Germanic languages and the Celtic languages, all group into Indo-European languages, which also includes the Persian language as well as a lot of various Indian languages like Hindi and Urdu. And so at one point there was a population that spoke Proto-Indo-European. And then as the language spread and diversified, we got more and more different languages until ultimately the language that we're all speaking now descends from that same language that Hindi does, even though Hindi and English are very different languages.
Right, Dapper. So coming off of, so you take the analogy, right? So you've got this nested hierarchy of languages, right? And we apply that same sort of principle to organisms, right? Whether crocodiles and alligators, or the Darwin's finches, right? Darwin's finches they aren't actually finches; they're tanagers, but there are different species, right? Most of them are members of the genus Geospiza magnorostris, Geospiza conorostris, but these nest with, so all the species of Geospiza nest together. They're sister to another genus, which is called Certhidia, and all of these are the Galapagos tanagers, and they nest within the family Thropidae, which is the family of tanagers, which is specifically the South American tanagers. They all nest together. And then Thropidae is sister to Fringillidae, the finches, so kind of ironic. And then they are then sister to this group, the Euphonias, and you just keep going from there. It's just different. It keeps incrementally going out these groups of passerine birds, and they're all very similar to each other, but they're getting more and more and more different as you're kind of inching away from where you started.
Yeah, and so
you go back to crocodiles as the example. There's a big difference between saltwater and freshwater crocodile.
Yeah,
but they're much more similar to each other than either one would be to an alligator, which is because any two species of crocodile are going to share a more recent common ancestor that itself would have been the first population that we could reasonably call crocodiles. And then, yeah, sorry, go ahead, Steve.
I was just saying, and Scott, you got to remember, these are all snapshots, right? I mean, 50 years ago, the word Google wasn't even in our vocabulary, right? Even before then, we had, you know, back when I grew up, nobody had heard words like people use like the word dude and bitchin' and, you know, every generation has new words being introduced all the time. And so that is an evolution of words. There's no specific time where all of a sudden we start speaking an entirely different language than it was 100 years ago. But I guarantee it, the vernacular we have now, if you were to go back into the 1800s and try using it, they would look at you like you're insane. They would have no idea what you're talking about, right?
I have a great example. I want to, can you give me like 3 seconds? I'm going to grab a book.
Run.
Yeah, go for it.
Well, while he does that, I just want to say, I don't have any problem with speciation.
Speciation is evolution, though. That's evolution.
Yeah.
And maybe we're getting confused about what you mean then.
All right. So as an example, sorry.
Wait,
hang on,
hold on
Dapper.
a second.
Sorry.
I don't have I don't have any issue with speciation. And is it safe to say that microevolution is speciation?
No, that would be macroevolution.
No, I'm saying speciation is not macro.
It is.
Yeah, it is.
Actually, the definition, so back in 1927, the terms micro and macroevolution were coined by this Russian entomologist named Yuri Filipchenko. And he defined microevolution as genetic change below the level of species, and macroevolution as genetic change at and above the level of species, thereby including speciation as part of macroevolution. Now, creationists have over the years done a very good job. They've run a very good smear campaign against macroevolution, trying to divide these because they have a sort of arbitrary separation point, even though creationists need macroevolution to work, especially if you look at modern baraminology. They have to have macroevolution work. They can't operate on microevolution alone.
Yeah, and I like the way you put that, because Filipchenko's if you look at his original definition, that actually, speciation is exactly that. It is at or above the species level. And so I think Jackson points out astutely, because he's, you know, the prince of profundity, that you know, when you have the creationist model, they have to have not just microevolution as they claim. They have to have macro as well, but they don't want to call it that. They play this word game to try to redefine it to something that has never been defined as by any recognized scientist, because speciation is a form of macroevolution.
Right.
So what's
I also
micro?
might be able to use as an example, and then we should probably get to Dapper's example.
Like all of the
sorry,
variations between us and between all the populations of humans on Earth, that would be micro.
Yeah, we're all
The
still
differences
human,
between
same
us
species.
and Aborigines and, you know, people in China, that would be microevolution. It's just a genetic change at the
Below the species.
Yes.
Yeah.
Let's stick with Darwin's finches.
Okay.
Sure. Okay.
Is that microevolution or is that speciation?
Well, there's both occurring.
That's actually
there's
adaptive
both,
radiation,
because
but yeah.
you have the original. So Darwin hypothesized correctly, I might add, that all of the Galapagos finches are descended from a single founder population. It turns out it was about 2.5-3 million years ago when the Galapagos Islands formed and these guys colonized it. This population then spread out into species, which then over time underwent a series of speciation events such that they are now different genera. You have two different genera of Galapagos finches. You have Geospiza and Sarthidia. So you had at one point just a speciation event separating their ancestors, but they've undergone multiple rounds of speciation events such that they're different genera and they have different species within them. There's Geospiza conorostris and magnorostris and septentrionalis and a whole bunch of others.
But within each of these species, we can actually, there's been I believe it's a husband and wife team who've been doing a long-term study. Yeah, they've been doing a long-term study, and they're also observing microevolution with changes in, sort of short-term climate cycles on the Galapagos Islands. And so they're watching each species also undergo microevolution without necessarily having any speciation events. Correct. And so they're seeing various changes in average beak shape and size over the course of these years. And that would be microevolution. So both are happening.
Oh, okay. So that's what I was looking for.
So Jackson, when you have, like, for example, if the founder population or the finches, right, whatever that population was, as that initial population grew and expanded outwards, they radiated outwards, what's called adaptive radiation. Each direction they go, where they go on the island, each things that they have to forest forward, things will change them so they get better adapted under selective pressure. So if they have a certain kind of nut, they'll have a certain kind of beak developed for that nut, or try to peck at a certain kind of tree.
Exactly.
So these are just minor variations in phenotype based upon ecological niches that they have for selective pressures. That's what the whole thing, which, by the way, he did predict would happen, but that's what adaptive radiation is. That's what
exactly saying. Yeah, the different food sources, different geographies. That's exactly correct.
Yep.
So micro, I can look at finch, like Darwin's finches. Their beaks changed. They're different changes.
They still might be able to populate with each other, right? They're not biologically isolated from each other.
They're still finches.
They're always going to be
finches,
yes.
Even with macro or micro evolution, they would still always be finches of some kind.
You never stop being your historical lineage.
Okay, we're gonna go back to that. Okay. Because I don't want to bring it up right now. I want to go over vestigial organs.
Okay. Real quick, can I just do this very quick example?
Oh, the analogy. That's right.
Yeah. So
sorry.
Going to the whole language thing, and I want to point out this is kind of similar to your having big changes from a pen to a pencil, even though man-made objects are usually bad analogies. So English has a history.
Okay, how about ape to a man?
Well, let me give the example. So English has a history that's about 1,200 years long, right? The first time we can really identify anyone speaking English, it's in the 800s. So this is some English from around the year 1000, and this is a story I know you know. So it starts: He queth, so thicha som mon haft twain suna. The queth se yngre to his father. Father, sell me min del min achte, theme je brith, da del de him his achte. Now,
what?
That is the first verse of the Prodigal Son.
You read that in Greek?
No, that was Old English. That's English. Yep. I would argue that's Old English from about the year 1000. So I want to point out, and this is not anything to your discredit, you couldn't tell what language it was at all.
No,
but
I couldn't.
Neither. If you just read it out to me, I wouldn't be able to tell what it was. Yeah.
But that's Old English, and if you've studied the evolution of English as a language, you can actually pick out how each of those words has either made it to modern English or been replaced by a different word or something like that. And all through the 1,000 years that separates us from the English speaker who wrote that down, at no point did English become some brand new thing that was completely different from what the previous generation spoke. So from generation to generation, everyone could understand each other all through that 1,000 years. But after 1,000 years, we're now at the point where, as far as you're concerned, English could have been Greek. And that's fair. It's extremely different from modern English. And so that's the same kind of thing where we can get, over time, without any break between generations, each parent produces offspring that are basically the same thing. But because there are these slow, gradual changes over the generations, we can get from something that is basically something you might buy at the fish market to humans.
Is that an example of micro or speciation?
Well, so it's a little tricky with language. Usually linguists use you
could say it's both.
Intelligibility. Yeah, linguists tend to use mutual intelligibility. So they would say, while at every step there was only microevolution, because remember, each set of parents could understand their kids and their grandkids and their great-grandkids and so on. Mm-hmm. eventually we get to the point through so much microevolution in English that essentially now we've had macroevolution occur where you can't understand any of it.
Can I?
You brought up an
screen share
Hang
real
on,
quick?
Jackson, go ahead. Go ahead.
Yeah, Jackson, go first. I just want to ask Steve if I can screen share for a second because I might be able to
Yeah, go
visualize
nuts.
this a little bit better.
I
go
think you might be. I think you're thinking in terms of different qualities of the processes that are happening.
I thought
would that be correct?
speciation and microevolution was the same thing.
Well, microevolution culminate or can culminate in a speciation event. Not
But
necessarily,
speciation,
but it can.
I'm sorry to interrupt. Speciation, you're telling me, is macro.
Yes.
Yes, genetic change at or above the level of species is the definition of macroevolution. So that therefore includes speciation.
Okay, I believe in evolution. We can get off now.
Wait, what? All of evolution?
Stream is over.
So are you saying that you would accept that humans and, say, chimpanzees would have a common ancestor?
No.
Then you don't accept evolution.
Yeah,
Well,
then
so-
you're just picking
So
and choosing.
there's
kind
two
of thing.
aspects to what is generally called evolution,
well,
right? There's
I think
the
that's kind of what I was gonna.
and common
That's what I was gonna kind of hint at, or that's what I was gonna try to get at. Was I think you're thinking that different, that qualitatively different processes are going on here.
Yeah,
at the
you can't have
micro
one. You can't
and
have
the
one without
macro
the other.
level, and that's not the case.
Yeah.
If I
You
can
have-
just show this, this will probably help Jackson. So I'm
Okay,
just gonna
sure.
Show this now.
I might be able to use it. What?
Yeah,
Is stream share going to work?
hopefully. So let me
It's
know. Can you guys see?
nothing. Oh wait. Oh, there it is.
It's nothing. No. Okay. So you have a different shape here. Now you have another
not
shape. Now you have a completely different shape. And I did squares on purpose. And now you've got a square. So you've made it from here to here, and then from here to here, and here to here. These are all small changes. Only one line
Well,
is being
Chesh-
added, but there's a big difference between this line and this square.
Chesh, I would actually say I think that color palette over in the top right corner would serve even
Yeah, 'cause it's
better.
Oh,
graduate.
absolutely.
Because you look
Exactly.
at the yellow, if you look at the center, what you consider the center yellow portion, you see where it melds to red. But at no point can you say it is no longer yellow. At no single line can you
There's no
say
line of
this is
demarcation.
not yellow anymore.
Right.
Right?
It's a
The
graduate. It's a
change,
gradient.
the gradation is imperceptible.
Right.
Basically, until by the time you finally get to red, you're like, okay, well, this is obviously red. When you
So
look,
here, let me do this for you.
See, here's the thing, Scott. You can't accept one without the other. It's kind of nonsensical. They just go hand in hand. One has to entail the other. Humans
Right.
and all other members of the ape species must have common ancestry together. We are apes. We have a common ancestry with all other members of the great apes and lesser apes.
the thing that it's not a qualitatively different process going on at the macroevolutionary scale than it is going at the microevolutionary scale. Either way, if for instance, I sent you the video Splitting Kinds. One of the examples I give, the very last example, is about the separation between saurians, which are manatees, and elephants. Well, the separation, while manatees and elephants are very different looking today, right? Elephants live on land and they have the long trunk and everything, and manatees live in the water. Their original differences were very, very minor. They were nearly identical in terms of their ambulatory capabilities. Like their hips are about the same, their body shape is the same, they're feeding on about the same type of thing. About the only difference is the ancestors of manatees, which are very similar to the ancestors of elephants, the ancestors of manatees were feeding on saltwater plants, whereas the ancestors of elephants were feeding on freshwater plants. Well, because they're feeding on slightly different types of plants, this is gonna have a drastic effect on their distribution, right? The elephants are only going to be able to be distributed in freshwater areas, while manatees, because they eat saltwater plants, are able to move about through saltwater, so they have a much wider potential distribution. And because of this, natural selection is therefore pushing them down separate evolutionary paths, right? So you end up with major differences, which were originally just very minor differences, but it's the same process happening. There's not a different process going on there. Do you see what I mean? And what Cheshire's doing is a very good example right now, because she starts with the very bright yellow on one end, which is getting progressively more orangey, but at no point is it orange.
Now at some point though, when she gets finished, the beginning and the end, those two different colors might be so different from each other, they would be considered to be different species because they're biologically, reproductively isolated from each other, right? But we don't know when exactly that line happens, right? There's no one hard line.
Well, here's the interesting thing. She could also do another line coming from the same point, but going in the other direction around the color wheel. So it gets progressively greener. And so that is basically, we start out as a single yellow organism, right? And then because of some split, whether it be geographical isolation, some kind of behavioral split, there's lots of reasons why these splits can occur. But once one does, the other one could get greener. And so if they both lead back to this very similar common ancestor, and so as they split and now they're no longer interacting reproductively, because there's no way for the genes to cross over, essentially, they end up going in different directions, and they become more and more different. And so that's the same kind of thing that we see with Latin, where if I read a Latin sentence to, say, a French speaker, who hadn't studied Latin, obviously, they wouldn't be able to understand me. And similarly, if I did the same thing to a Spanish speaker, they wouldn't be able to, even though they both descended from populations who would have originally been perfectly well able to understand it. And so the same thing happens with organisms. As time goes and once the speciation split occurs, those two populations, presuming they survive, will always be getting more and more different. And they themselves might speciate after that, creating, say we get two species out of one speciation event. Then later on down the line, those two species are now much more different to each other than their common ancestor groups were. And then those groups could also speciate. So we get even more differences. So that's why we can get things like, we can say that alligators and caimans are pretty closely related. And so are salt and freshwater crocodiles. And we know that alligators and caimans are closer to each other than they are to crocodiles. And both crocodiles are closer to each other than they are to alligators and caimans. But there was still a shared population ancestor between them that still would have been basically a crocodilian. It wouldn't be a crocodile, but it would have still looked very much like an alligator or crocodile. Would have had those big armored scutes on its back, nice sharp teeth. It would have lived in the water, had that vertically flattened tail, all of that. You would look at it and you'd say, I guess it's not an alligator or a crocodile, but it's clearly something similar. And then minor differences occur. So maybe crocodiles continue to have a larger portion of their diet as fish, say. I don't know this for certain. I'm just sort of going with an example. And so they keep a narrow V-shaped snout. And alligators start doing more predation of land animals that are coming to the water's edge. And so they get this nice broad snout so they can more easily latch on to medium-sized prey. And so we're getting that differentiation.
Here now is grab
That's a very good
these
example.
colors and put them all side by side. And you can tell me if you think that they're the same color of where we started from to the center to all the way to red.
And imagine these colors going around some kind of boundary, like a geological boundary, Scott. Like, for example, a mountain, right? Mountain range. You have one population going to the right of the base of the mountain. You have another population going to the left of the base of the mountain. You have one going up the side of the mountain, right? Each one is going to run into different ecological changes, right? The different temperatures, different food sources, different predators, predation, rainfall. There's going to be numerous things that are going to change what that species, that population that goes that way has to do in order to survive. So if you have a population that has diverged through a geological reasons called allopatric speciation, you look at one population that went to the right, one to the left, one went to the top. All those different populations are going to have very fundamentally different phenotypes. They're going to be different, and they might have been so great of changes that even if you brought them all together again, they would not be able to make. Even though they came from population.
And you can observe this sort of differentiation based on environments in things like ecotypes. So if you take like a plant, for instance, and you can look at this one species of plant in like the Sierra Nevada and in a Florida swampland and in Washington State. If you look at it in all these different places, it's going to have a particular set of adaptations for each place. It's probably going to have water conservation, mutations and adaptations in the Sierra Nevada, and maybe warmth adaptations or whatever, or to keep out the cold in Washington State.
Yeah. So does that
Well,
make
etc.
sense?
You can
Scott,
observe things like that.
Well, just to be clear here, at no point would you ever say all these are the same color, correct?
Right.
And you just watched me go from one color to the next. But if I were to take red and let's say I was doing a painting and I decided to replace let's say it's daytime and I'm just painting a field and it's the day, so I've got the sun in the sky. The entire picture changes if I remove all the yellow and replace it with orange. They're not the same. They can't be they're not interchangeable that way. They become different things, even though you did just watch me go all the way from one to the other, where you can't tell me where here did it become orange and where here did it become red.
And you can actually do this with certain organisms in the fossil record. With larger things like vertebrates, you often have a very, very patchy fossil record, such that we may find some manatee fossils in the Eocene, but we don't find manatees later until the next set of manatee fossils until like 10 million years later.
Yeah,
right? That can happen with bigger things. But with smaller things like diatoms and forams, these little protists who build these shells, you can look at these beautiful, eloquent core samples where you can see what is nearly their entire evolutionary transition over a few million years.
I just don't want Scott to get a wrong impression. So what Cheshire is drawing, this is what's called anagenesis. Anagenesis is more of a linear type speciation, when most speciation is called a bifurcation, or what generally known as let's think of it as splitting, right? You have a population that splits into two or more populations, right? Those are generally how you have evolutionary things. When you look at a phylogenetic tree, you'll see these bifurcated patterns repeatedly.
Well, I could also make this far more complicated. I could take this yellow, like Dapper was saying, go the other way and go
exactly.
green. I could then take the orange and go back to yellow or to red. I could then take the red and go all the way to purple, or all the way around to green again, and I can just all loop them all in all the way around, and you can see the.
Let's get Scott's. So far as he catching up with all this, we've kind of thrown a lot of stuff at him.
Yeah, you are.
Sorry about that.
But I was listening.
So do you have questions or why you think this is perhaps not related to what's going on in biology, like by analogy?
No, you actually, it was, you gave several examples that were good examples.
Okay.
And I understood them. I didn't understand all the words you said. But
sorry, I do that.
For the most part, I understood what you meant.
If I ever say a word you don't know, interrupt me and ask me.
That's about 90% of your vocabulary, Dapper.
Oh, Steve, you're not wrong.
We only got a 2-hour show.
All right.
Okay, so speciation is macro.
Yes.
It is a form of macro.
Okay, so let's not go to Vestigial right now. I want to stick with this.
Okay.
Science says we evolved from apes, gorillas, chimpanzees, whatever you want to call it.
Hold on. We didn't evolve from any modern ape.
Yeah.
But the common ancestor between, say, humans and
we're still modern apes. I mean, we all.
No, I have Homo habilis, 2.8 million years ago.
Mm-hmm.
Okay.
I have Homo erectus, lived 1.6 million to at least 250,000.
Mm-hmm.
And then there was the Cro-Magnon, but I guess
that doesn't really
now they're not.
Yeah,
they kind of threw that aside.
You're looking at Homo habilis.
It's just modern Homo sapiens.
Yeah.
Yes, Cro-Magnon is basically just Homo sapiens.
Yeah.
So, so there's basically been 3.
Well, no.
Oh no, there's a lot more than that.
Homo erectus, Homo habilis, Homo florensis, the
Homo naledi. Yeah, there's a lot more of them. These are just kind of some of the big ones.
Let's just stick with humans. Okay. The first human was around 6 million years ago.
There was no such thing as the first human.
So there was a population which split And one lineage led to what are called the hominins, which was the human lineage, and the other led to the chimpanzees, right? The chimpanzee lineage. And right. But there was no single first human. There was no first human born. It was just a slightly more human-like population.
Okay, so how about this, Scott? So there was a population called Paralithicus catalonicus. It's about 12 million years ago. Okay, from that population, there was divergence. Some of them became pan-troglodytes and chimpanzees. Others went to other forms of hominidae, which is going to eventually lead into us. But that was the common ancestor. That was, I think, they found it in Spain.
Steve, I think you're thinking of something that's karyotypic of the common ancestor of all of hominidae.
All of hominidae, yeah. Mm-hmm.
So that group would have split into orangutans on one side, then gorillas, chimpanzees, and humans on the other side.
Right.
Then the population on the gorilla, chimpanzee, human side, the gorillas went one way, and then the chimpanzees human group went another way.
Yeah, so exactly.
Both of those groups split into the two gorilla species that we have, and then genus Pan and the hominins on the other side.
Yeah, the last one for chimps and us was Pan prior, which has not been discovered, if I remember correctly.
Yeah, that's a sort of hypothetical name.
Yes.
Although I don't like that it's used Pan, because it shouldn't have the same.
Sahelanthropus is approximately what you would expect. It's got both like dental and cranial similarities, both with the Pan lineage and the Homo lineage. And that's kind of why it's just barely on our side, but it is on our side. That's like the earliest hominin.
So Scott, those go ahead. Because those were human populations that no longer exist, right? They went extinct. They did not survive.
But I want to see what Scott has, because I feel like he has a question.
I have several.
Awesome.
So I'm at a fork in the road.
Okay.
I can, like a Y, I can go this way or I can go this way.
Mm-hmm.
Right? So you have chimps and whatever and gorillas and whatever. What is this right here? What's this?
So the intersection, there's a couple things. One of the things you have to realize is the way that we make our little, you've seen the little family trees, right? Because in the fossil record, it's always very hard to say precisely whether a single fossil taxon that we have is directly ancestral to a modern one. The way that we do it is we simply conclude that all of these are tips of the branches. Now, we might actually be wrong and say that one of these tips might actually be the little node at that intersection right there. It could be the case. But what is always the case at that intersection is that what we have is a population of organisms that will, for some reason, split into either rapidly or slowly, whatever. So if you were to look at the little node there, if you were to somehow be able to go back in time and find distinctly the population of apes that would give rise on one side to chimpanzees and bonobos, and on the other side to all the humans and their close relatives, it would have looked, it probably would have been a mostly tree-dwelling ape that sometimes came onto the ground, maybe to forage for fallen fruit or something. It probably occasionally ate some meat. It probably hunted some other primates or maybe some, arboreal rodents or something. It was probably about as hairy as most of the great apes are. It probably didn't knuckle walk, actually. It probably was getting around mostly by awkward bipedal motion on the ground, but staying in the trees most of the time. The reason I say that is because gorillas and chimpanzees knuckle walk in very different ways. So the current idea is that they actually both developed similar, but not the same kind of terrestrial locomotion separately, which also would have meant that the common ancestor of humans and chimpanzees probably wasn't doing that. And so then on one side, we have a population that lives in what's now sort of the Ethiopia area of Africa, which at this time, about 6 million-ish years ago, was drying out. And so jungle was being replaced by grassland. So all these adaptations to moving around in the trees weren't very helpful. And so there was increasing pressure to become better and better at walking on land. And so this group of apes then adapted to become bipedal, because primates already have a tendency towards bipedalism. If you look at gibbons or a lot of lemurs, they don't. Yeah, they tend not to walk with all four limbs. They actually tend to only walk with the hind two, only occasionally, or hop, yeah. And only occasionally do they actually use their front limbs to support them on the ground. And it's usually only when they're doing something in particular. Like
I think dogs might be a good example here. Can
Well,
you
I just-
tell me what? Oh, sorry. Finish. Go ahead.
Yeah, I'll just go ahead. So that's sort of the expectation of what we would roughly see if we could see this population. But because we can't ever say for certain, with maybe one or two exceptions, that specifically this species that we found, we know it was the ancestral species. We can say it meets what we would expect to see. And one of the other things that you have is this population that exists would have had other populations that were related to it nearby, but that didn't leave descendants. That population, for whatever reason, would have gone extinct. So chances are what we would have found is probably one of the sister groups rather than the exact one, because there would have been a lot.
Like Homo naledi.
Right.
Like the number of circles I had in that picture.
Or Neanderthals and
There was a lot of circles
Denisovans.
in there.
Right. Yeah. If you had looked at humans not that long ago, geologically speaking, there would have been about 4 species of Homo coexisting. In fact, at some point you could go back and get to 5 of genus Homo, all fairly closely related. It's just that all of them but Homo sapiens ended up dying out. So now if humans were to go on to speciate, like maybe some Elon Musk gets his wish and humans colonize Mars, and because that's such a hard thing to do, people don't really travel back and forth. Well, humans would evolve to adapt to a life on Mars, and eventually they might speciate. And if for some
A good
reason,
example of this is if you change gravity, right? If you were to change the gravity on Earth, because that's a kind of a big thing in a lot of sci-fi. If you were to make the gravity heavier on Earth or wherever humans are living, humans have a very, very, very hard time surviving. Breathing becomes difficult. The way your joints are and the way they move becomes incredibly difficult because you have small bones and small muscles that are adapted perfectly for working in the gravity that we deal with. But if you lessen it or you heighten it, you start running into problems. You see that when people spend so much time on the space station, they have to have additional exercise machines and so they their
reason they come
muscles
back
don't start
out of the chair.
deteriorating.
They're having huge physiological changes. Yeah.
yeah. So does that answer your question of what is at that node?
And well, think about it this way, real quick, Scott, and then we'll get back to you. Is that if you have 2 nodes on an apple tree, right? You got 2 branches, and at the very end of those branches, you've got one apple, like let's say a red apple, and then some other branch, it's got a yellow apple for some reason on the same tree. You have to, just by sheer reasoning, they have to come from a node that's similar to both at some point. There could be no other option. They have to have a common node. And in that common node, you're going to find the similarities that allow one apple to be red, one apple to be yellow. Same thing with populations. If you look at 2 divergent populations, if you look at their genetics, and you go back to whatever founding population or whatever node they came from, they have to have a commonality to them. There's no other way. And so when we see 2 populations, we go, okay, there has to be a common population that existed to both these different types of species at some point in time. What that species was, they don't get into details with. There's no way to really detail what it was, but they have to know that there was one. Does that make sense?
Right.
Yeah, it makes sense. I would like to say that Dapper was giving me was explaining in great detail of what might have or probably did. And I did catch that he said probably 3 or 4 times.
Yeah.
But we don't know. Now, on that,
Well, what do you mean by
there's
we
a lot
don't
of
know? What do you mean by we don't know? Because you can have probably and we still know. You don't have to have certainty to have knowledge. Right?
All right, I'll give you I'll give you an example. He said, there were apes or gorillas. Pick an animal. I don't care. Comes down, and they have to be better at foraging for food on the ground and eat meat and walk and climb back up, and they had to they probably had to be more acclimated to the ground.
Well, there's a methodology for determining this, right? There's a set of things that researchers, anthropologists in particular, and geologists and lithologists all looked at, which
Well, here's my
led
question,
them
and you
to
can
this
answer
conclusion. Sure.
Here's my question. How do you get all of that information from a skeleton?
Okay.
So you're not just looking at the skeleton. That's part of it. If we're talking about hominins, for instance, we can see if you take the hominins that we're aware of, like you mentioned Homo habilis, Homo erectus. What you see, if we're going from things that are much closer to our common ancestor with chimps like Sahelanthropus is they have a lot of differences. They have more differences. They have a more prognathic face. We have a slightly different dental setup. We have different. They weren't as bipedal as us. They're more hunched over and all that. But over time, you see these changes. You see changes in the teeth, changes in the shape of the face. It's less prognathic now. It's much flatter as opposed to chimps. You can see that the spine is shaped in a certain way. You have the foramen magnum, which in the more or the less bipedal apes is more. It's more forwards, right? It's more forwards in our skull, isn't it? And it moves. It has moved
more forward.
Sorry, it's more forward. It was more back, and it has moved more forward. And you can see this in our fossils. You can see how it moves.
Well, you can probably use,
but
as a bit of an example, is how much do you think humans have changed based on certain inventions? So farming, for example, has changed a lot of what humans are able to digest.
Well, I mean, yeah, you're right, and that does also affect. If you look at like the history of Europe, you had
social and economics also are a factor that have to be taken into consideration.
The lactase persistence, but also with it's not just the fossils. You're not just looking at the fossils because you see this change, and when you compare the fossils, you can see that they are changing. Right, they're changing in a certain way. They're becoming more bipedal. They're walking in a certain way. They are able to eat certain foods because you can look at the teeth. You can see their shape. Are they eating more meats? Are they eating foods that they have to crush, brittle foods? Are they eating more fruits? You can tell a lot about an organism based on its teeth alone, right? Yeah. And so they can run all these sorts of diagnostics on our teeth and figure out what we were eating at certain times because there are other. You have the Australopithecines. They were eating more.
I don't know what that is.
Well, so before you have Homo, you have our genus Homo. Prior to that, the ancestral genus to Homo is Australopithecus. Not any particular species, probably, but you have the genus Australopithecus. We're descended from them. Right? That's getting closer towards our common ancestor. You have Homo, so you have us, and then you have Homo erectus a bit further back, Homo habilis a little bit further back. You have Australopithecus, and you're getting closer towards the node with chimps, right? And so we can see these changes are occurring, but we can also look, and when anthropologists, they have geologists with them, and they're looking at the environment that these organisms were living in. The geologists can look at these environments, look at what type of rock is forming, look at what, if you have palynologists, you have people who are looking at the pollen, they can see what kind of trees were living in this area, and you can use these to infer it was becoming drier. You can also use things like oxygen isotope ratios. So you can see, was it becoming cooler and wetter? Was it becoming warmer? These
from a skeleton.
No, not from the skeleton, from the rocks. From the rocks that they're being found in, the matrix that they're being found in. You can see the
environment by looking around at
There's a whole holistic approach. It's not just one thing, right? And you can tell a lot from a skeleton. God, I mean, Mary Schweitzer determined that the T-Rex that she was working on by looking at the osteocytes had been given birth. That's pretty impressive.
I think I have a non-technical example that might help here.
But, well, okay, and then, I just want to ask, so it's, you understand it, they're not just looking at the skeleton, right? They're looking at the whole of the site that it's found in. They're finding the pollen from the trees, the fossil pollen from the trees that it lived in, possibly other animals that it lived with, like boars or antelopes or elephants. They can see because of the rocks, not just the skeleton itself, that the environment's changing. You get that?
Yeah, that's not something that I even thought about.
So does that sort of change the way that you are perhaps going to consider?
Yeah, absolutely.
Okay.
Yeah.
Cool. Okay.
All right. And this is a fairly. So I noticed one of the things that you said was I said probably several times, and that's fair because one of the things is science is always cautious about making absolute statements when there is some possibility of being wrong. So like science could say, yeah, the Earth is definitely a globe, and it's also definitely way older than 6,000 years old, but it's about 4.5-ish billion years old. It's not, we can't say it's exactly, 4,500,000,000, whatever. So here's an example that you could use just using like a family, right? So I did a DNA test a while back, and it turns out that about 2/3 or so of my ancestry is from Ireland, but specifically County Cork, which is a county on the west side of Ireland, right? But let's say I find someone, and because this was an Ancestry.com, they'll tell you these things. And let's say I find someone who turns out to be like my 7th cousin. And so we meet, and we actually have like a sort of vague family resemblance. And so we check our genealogy data, and it turns out that we can both trace our ancestry back to 2 men that lived in the same city in Cork, but we can only do it back like 4 generations. We're not quite back to 7, which is where we would like to get. We'd like to see our common ancestor, because we probably have one. Actually, we certainly have one. But we think it's probably in this particular city in Ireland. So we go to the city, and it turns out that because we know about how long a generation time is, we can find the graves that are about the right time for our ancestor to have lived. And so if we ignore the ethics about exhuming bodies just for, funsies, we'll just pretend that that's not a problem. If we were to exhume some of those graves, and let's say they're unmarked, we could find a few skeletons, all of which, if we did, some forensic reconstruction or measurements of our own skeletons, whatnot, we could find the ones that look the most like us. And one of those, say there's 3 of them, one of those is probably our common ancestor. It's probably the actual, many times great-grandparent that we have in common. But it also could just be someone closely related. Like maybe our common ancestor was lost at sea or something, and they were never buried. But we can still say we have found some things that are at least very, very close to the common ancestor that me and this other guy share. Because we've done the checks, we know we have these genetic markers in common. We know where those genetic markers are found. We can measure, parts of our body, like our skull shape or whatever, limb proportions, and see where we're similar in that way, where we might be more different to people who are less related to us. And then, but we just don't quite have enough to say, yes, this is the skeleton of my 7 times great-grandfather. But we can say this skeleton might not be him, but he was probably very closely related because we share all these skeletal features, and we trace it as far back as we can directly with history and genetics. A similar thing occurs in the fossil record, where we can say, okay, we know evolution happens. We know it can result in really large morphological change. And so we know about how long it's been based on things like, it's not technically mutation rate, but it's a similar rate that's based on how often mutations end up getting...
Substitution rate?
Yeah, it's sort of the substitution rate or the phylogenetic replacement rate. But we can measure these things in real time in real populations. And then we can work back, okay, so how long ago did these 2 populations share an ancestor based on this mutation rate? And then we can look in the fossil record at that time and in the places where we think it's likely that they go back to. And we can find organisms that, while they might not be the exact ancestor, are what we expect. And one of the more famous ones, although it's not actually exceptional, it's just famous, is Tiktaalik. So Tiktaalik is a very fish-like, but also sort of amphibian-like organism. It had scales, but it had 4 limbs, but it didn't really have fingers. It had something that was sort of mostly a ray, but
it has ribs and lungs like a amphibian.
Right. And so, but the thing is, the reason that these people were digging out in like far North Canada for 3 months a year, because that's the only time it wasn't frozen, and they did this for years, it's because they expected to find that. Because they knew about how long it had been since, say, frogs and humans shared a common ancestor, and they knew that there was exposed strata from that time. They knew that if they were right, that it would have been very fish-like. It would have been in sort of an area where we're transitioning from the sea to the land, so it's sort of a beach area. And they knew that the rocks in that area were from about the right time and the right environment. So if it's true that humans and frogs share an ancestor, and evolution is correct about when this happened on the basis of our measured rates of genetic change, then that's where we expect to find something that, even if it's not the actual ancestor, just like that skeleton I find might not be literally my great-grandfather, it's at least closely related. And
yeah,
so,
and not only did they find one, they found hundreds, if not thousands of them.
The thing is, the reason I say it's not exceptional is because most paleontology digs are based on this, because you only have so much funding. So if you want to say, I want to investigate the evolution of, say, I don't know, Tyrannosaurs, right? We say, okay, well, we know we have different groups of tyrannosaurs in North America, but they're all very closely related based on, you know, how similar they are. But we have more distantly related and more diverse tyrannosaurs, and this is in a broad category, in China. And we know based on things like how fast we see the skeletons change that we're probably expecting mid-Jurassic. So we go to China and we dig in mid-Jurassic rocks and look what pops up. Things like Dilong, which is identifiably Tyrannosaur, but it's also very similar to the other non-Tyrannosaur but related to Tyrannosaur dinosaurs. It has longer arms, it's got a smaller head, it has much more primitive metatarsal bones, which is sort of the bones in your foot that make up the long portion of your bone past the ankle until you get to the toes. So it has all these things that are much more similar to the other related dinosaurs. But we can make that prediction. And that's how paleontologists actually get funding, is they have to make these predictions. And if they were never right, the funding would start to dry up, because the people who are paying you to find the primitive tyrannosaur aren't getting it ever. So why would they keep paying you?
Scott, imagine drilling for oil. As a matter of fact, there wasn't an actual oil place that called. There was a creationist oil company. I believe it was called Zion Oil. They actually did not have a evolutionary type model, which would be, you know, deep time geology stuff model, and they kept drilling dry wells. They couldn't find oil, and as soon as they eventually they had like 3 dry wells, and of course that's a lot of money that they've wasted, and they had to go to a model that we knew worked, which would be modern geology. And by using modern geological understandings of where we would expect to find oil, guess what? You know, then I think they had one. Did they find eventually one well? I don't remember, but
they eventually started finding oil basically after they gave up trying to use the Bible as their book.
Yeah, exactly. So I mean, these things have to have predictive ability, or else, you know, people are just going to be like, oh, well, they don't do any good. You know, we're not going to throw money away for nothing. Now, you might have individuals that believe in some kind of woo that, well, hey, I'm going to buy this, you know, necklace hoping it does something. But when you have when you have a multi-billion color company, they're not going to waste money on that. They're going to be like, hey, look, if you can't get results, we don't care what you believe. We're not going to fund you. Period.
Yeah, the continued success of oil industry in the world depends on evolution and the geological time frame.
Creationists don't have funding for a reason. They have no funding besides people that want to donate to them, because they have nothing to ever come to fruition about any kind of predictions they can make to do anything. There's just nothing. There's nobody profits off that. Yes, it's sorry that it comes down to money, but there's a realistic aspect to that. I mean, seriously.
Hey,
if it makes it feel any better, science struggles in the same way. There are sciences that could probably be very, very lucrative if
they had results.
If they were funded properly, but they're not funded properly because they're not viewed as lucrative.
Or
my field. Sorry,
over on my channel.
Yeah, exactly.
Sad day.
Yeah, you might have a field in science that is, as of yet, has not produced results that might possibly. I mean, they're speculative.
Studies in algae actually are really huge. So, like, would be very, very profitable, but that it doesn't happen, and it should be, but it's not.
Go ahead. We're getting really close to it. Scott, so go ahead. Yeah, sorry.
Oh, it's my turn?
Yeah.
Oh, thank you.
Sorry, you forgot he was here.
I didn't forget.
Okay, so what is the chart called with the different strata?
There's a few different, like the geological time scale, or a stratigraphic column, or something like that.
Where it shows, you know, it's this layer is this many years old, and this is older and older and older, right?
Right.
Okay. And Steve, you brought up, well, you know, if we go into this strata, we know that, you know, we found XYZ, you know, these bones here. So, you know, let's stick to that strata.
Right. Index file.
My question is, could you just give me like what's the deepest strata, and just could you give me the deepest strata where we find that stuff?
Where we find fossils?
Yeah, where we find fossils, or just fossils?
Specifically.
There's a site in Greenland. I believe was it Ishua? I think that's how you pronounce it. Where there are some fossils that are claimed to be like 3.77 billion years old. They're like stromatolites from near hydrothermal vents.
Okay, but what time? What
That's
strata?
like 4 billion years
Is
old.
that
That's
far into?
Archean. That's Archean eon or early Archean eon.
So what's the top layer called in the geological chart?
The top layer. I mean, you have like the Cenozoic, which is what we're in right now, and then you go down and you have the Mesozoic. Then you go down further, you have the Paleozoic, and then you have the Proterozoic, and then the.
Okay, that's what I was asking
Well,
earlier.
yeah, well, those
Okay,
are
at
also
the
broken
very
down
beginning.
too, right? You have various epochs, like the Holocene. Holocene? Epoch?
Well, it's not all. You can't. It's not all in one place.
Right, there's no
because of
contiguous
how,
column, correct.
yeah, there and this is part because of how like Dapper was talking about. You have mountains forming and you have continents colliding with each other, and this can destroy and metamorphose different sediments and things so that they're lost to time in certain areas, but you can pick them up in other areas.
Remember when I asked the crust, how deep is the crust?
Okay.
And then I asked, do we find the fossils in that, or do we find them lower? This is what I was talking about. The Paleozoic or the whatever Zoic, and you just named all those things. That's what I was looking for an answer from the geological chart.
Mm-hmm.
What do you mean?
You said the first.
What we're in now?
But I'm not sure the actual question though.
Yeah.
Okay, so I know there's a geological chart, right?
Right.
Where someone did back in the 1800s, I think, and they were on the side of a hill or what mountain or whatever it was, and he saw 2 feet of this kind of rock, and then 2 feet of this,
Right.
Okay.
That, what is that called?
That's chronostratigraphy. So that's basically what you do is you're looking, because you're not going to find all of these in one place. What you have to do is you can't really use.
It's not about place. I'm asking, if you go this far in the earth, it's this old.
Yeah,
and then
you can't do that universally.
Like that.
Yeah,
you can't do that universally
geography matters to geology.
Yes, because of the earth, because the tectonic plates have collided and separated, and some have subducted. This destroys sediment in different ways, which is why you can't find all of them in one place.
And also, I think it needs to like recognize that you might have, like, for example, like Triassic and Jurassic, you might have physically higher a Triassic layer, even though that it came before the Jurassic because of geological conditions, right?
Yeah,
so
Cretaceous could have been laid down at a lower elevation, or the Jurassic could have been uplifted.
Yeah, exactly.
How old is the Jurassic?
Earth moves.
Well,
the Jurassic is 200 to 150 million years.
So 200 million years ago is Jurassic, right? So let's say you go a little bit before that, and you look at the Jurassic. So Jackson, that's when the first mammals started coming about, right? Give or take.
Jurassic.
Yeah. So about 300 some odd million years ago.
Yeah, basically.
Yeah. So you have these first mammals, right? So we're going to find them if we say, where are we going to find these mammals? Let's look in what the geological strata for the Triassic is. We're not going to find mammals below that. We're not going to find that in the Denovian type strata. It's just not going to happen, right? Because that would actually disprove evolution. We'd be like, how do we have a mammal that's that exists when you're just coming out of the Cambrian for the most part?
Okay.
So what is your follow-up question then?
All right. So the follow-up question is the Jurassic.
Okay.
Is it fair to say that's where we find dinosaurs?
That's one of the layers, yes.
So we find dinosaurs starting in the late Triassic. They become more and more common by the Radian stage, which is the last stage in the Triassic. They're starting to take up a lot more of the sort of the population of land animals. Then
Triassic.
Okay. And then the dinosaurs are dominant all the way until the end of the Cretaceous, at which point only a small group of birds survive from the dinosaurs.
Okay, you know what?
So,
let's make it easier. What layer do we find the oldest Homo?
So you said about, what was it, 2.8 million years ago? Is that what you said earlier?
Because
that sounds about or 3 million years, something like that.
Yeah, 2.8 million.
Yeah,
Is-
2 point.
All,
So they-
all humans are going to be in the quaternary. Every one of them are going to be in that period.
Oh yeah, that's true.
Yeah, you're not going
what's
to
it
find
called?
anything. Quaternary?
The quaternary.
Quaternary, yeah.
Yeah, you're not going to find any humans,
Yeah, I think
below that.
the quaternary is like 5 million years ago to
Yeah, something
like
in the
10,000 years
Cenozoic.
ago.
But I don't know
Okay.
that.
Well, it's still,
So-
it's still present because the Holocene is still part of the Quaternary.
Yeah, so you,
Okay,
you,
yeah,
you have,
you're
you
right,
have,
sorry.
you have these. You got to remember these things are broken down by era, epoch, period.
Anyway,
Do you
So-
want me to spell it for you so
Yeah.
you can look it up later? Okay, yeah, it's QUATERNARY.
Thank you.
I figured that one would be a little bit difficult, so I googled it.
Yeah, I mean, if you found a
So-
human in the Jurassic layer, that'd be a big problem.
yes.
Oh, that would be.
So what's
And
your question?
it's called quaternary.
Quaternary is the
Yes,
period.
yes.
The era is Cenozoic. The period is quaternary. The what is it?
All right.
Eon is Holocene.
I think the
Well,
stage.
Quaternary. We're all together with our little fluffy bunny hats on and mustaches, and dinosaurs with top hats,
Mhm.
and
Wispy
so
hair.
we know that there are humans
Sure,
in
I
the
found it.
quaternary
Mhm.
because they've already found them. So we're digging.
Okay.
We find a skull. We find fossil
Mhm.
bones.
Okay.
I don't care. Pick a bone. I don't care.
Pick a bone.
All right.
Pick a bone.
I picked-
I found what we were looking for, by the way.
Circle bone of the first digit. That's the bone I pick.
There you go. Oh, cool. All right.
So this
all
is
right.
what I was
So-
talking about. I
I see the
couldn't think
fifth.
which was
All
which,
right.
but so we're right here in the
So-
Holocene epoch, which is part of the quaternary period, part of the Cenozoic era, and the Phaeroneozoic eon. And then as you go lower,
Steve's getting way too complicated, my dude.
Steve.
No, no, I
me?
like this.
Okay.
have you
so-
listened
so
to
Scott-
Dapper? I'm at least showing him nice, pretty pictures here.
No, this is good.
I know young Earth creationists like pictures. So when we're talking
Okay.
about the Jurassic and Triassic down here in the Mesozoic, way earlier than we are way up
All right.
here.
Let's just stick with
Okay.
quaternary,
Quaternary,
Yeah.
this is here. Yeah,
Quaternary is
we're
here.
here.
Cenozoic, quaternary.
Mm-hmm.
We're in a spot where they have found bones before. We
Okay.
dig. We find bones.
Mhm.
Okay. Now, do we automatically say Homo erectus or Homo
No.
habilis or
No, it has to have the features diagnostic of that species
No.
for us
All right,
to
let's-
classify it as that species.
let's say we find a complete skeleton
Okay. Sure.
and we say Homo habilis 2.8 million, 2 million years ago.
We
He's
don't.
2
We
million
don't.
years old.
Well, that's too old. But ignoring that problem, because I realize you're just picking a number. So
Yeah,
when
all
we
right.
find the skeleton, the first thing that happens after excavation is we take very careful measurements of as many things as we possibly can. You actually break out the ruler, the calipers, all that stuff, right? And then we
Okay.
compare it to very well-defined lists of characteristics that basically identify a species as what it is, right? So we'll say, okay, do these measurements fall within the range that's been already established for, say, Homo habilis? And if the answer is yes, then at that point we can feel safe in saying that this is an example, this is a specimen of Homo habilis. But if the answer is no, then we have to check other species. And if the answer is no to all of them, then we have likely found a specimen of a new species, at which point a new name has to be published for this species.
Okay.
Yeah. So
Forget about the whole skeleton. We find a bone. Okay.
okay.
How do we know how old the bone is approximately?
So there's a number of things. In some cases, if it's fairly recent, we can directly date it in a few different ways. One of which is carbon dating. But if it's much older than that, then what is generally done is there's been this big long project that's still ongoing to date fossil-bearing layers, and usually you can't do it directly. But what you can do is you can find the nearest volcanic layer going up and the nearest volcanic layer going down. And because you can reliably date igneous rock with various radiometric dating, you can then say the rock sandwiched between them is after the lower igneous rock was formed and it's before the upper igneous rock was formed. So that's why when we give date ranges for these periods, we always say they're from this to this. So when you see a new fossil described, they'll say this fossil came from 2.4 to 1.9 million years ago, because
plus or minus like a couple million years.
right. So and that dating can get more and more exact as we go closer to the present, simply because less of that will have eroded. So as a general rule, the farther back in time you go, the bigger the date ranges get, simply because you're less likely to find closely associated igneous rocks, just as a result of weathering and subduction and all this stuff. So generally what happens is you check the strata, and then you check dating studies on that strata that are based on associated igneous layers. And so then you get a date range. And it's very hard to say within that date range exactly where you are, although you can sort of assume that the lower you are in that stratum, the older between that date range it's going to be. But even then, that's not something you can really say with high confidence. You should generally just give the date range from when the previous igneous layer was formed to when the subsequent one formed.
Jess, you want to read the super chat real quick? Because we had a super chat. Yay!
Yay!
Science super chat.
Yeah,
Yay!
The Dahlia for $4.99 said, if you need visual showings of geoscales, you can show him the slide show Colton made for me on my vid, #geological history 2. So I'm in the middle of pulling that stuff up so I can share it in chat.
All right, so what else you got for us, Willie? Hayes?
Okay.
Well, does that answer the question?
Yeah, it does, but I want to tie it into my questions here. So you've got carbon dating, you've got potassium argon dating, you've got uranium to lead dating,
Rubidium, strontium, loads and loads of different date methods.
Yeah. All with different degrees and whatnot. Okay.
Correct. Yes.
First, let me understand half-life.
Okay.
So if carbon dating, when I looked it up, there was a 14 and then a C. Is there a difference between that and then, or saying C14?
No.
So C14.
So what? Oh,
All right, hang on. We're in my territory now. All right. So,
hey, it's my territory too, Steve.
Yeah, but
hey,
right,
I took chemistry also.
I know.
We won't
this is all easy stuff. All right, so,
I do an art.
But
Chest knows colors.
Yes.
Okay, so one thing you got to remember, Scott, is that the carbon 14 is called an isotope. Okay, so an isotope is a different type of carbon. The carbon has a numerous amount of isotopes to them: carbon 12, carbon 13, and so that's just one type of carbon. And there's different types of structures of carbon, right? You have graphene type. You can have a buckyball. You can have diamond. These are all different types of structures of carbon. But the isotope itself, carbon 14, is radioactive. And what that means is after a period of a certain time, that half of that material is going to be gone. It's going to just be decayed. So when you say a half-life, that's the amount of time that it takes for a specific amount of a isotope to no longer be that isotope any longer.
So,
Yeah,
so the C12. So C14 is radioactive and it's a type of isotope.
Yeah.
Yes.
Mm-hmm.
And obviously, at its genesis or beginning, it has 14 carbs, and half-life it has 7.
Well, it has 6 protons, and then it has 8 neutrons.
Yes.
So when you add those two numbers together, you get 14. So carbon always has 6 protons.
Yes.
That is what
carbon
makes
is
carbon.
Yeah, you got to remember on the periodic chart, Scott, that the what makes carbon is the fact of the number of protons, right?
Right.
So a carbon is always going to have 6 protons regardless, because that's what makes carbon, right? If it had 7 protons, it would be the next thing up on the periodic chart, which is going to be nitrogen. So when you have something like nitrogen, like nitrogen 14, and all of a sudden you have in the upper atmosphere, it can bombard with a neutron. It'll actually produce a proton and carbon 14 in the atmosphere. This is the cyclic thing that it keeps on renewing itself in the upper atmosphere, is constantly producing carbon 14. And so when you look at the number and you see like 14, that's just tell you the atomic weight per se. It's kind of it's number of nucleons is what it is, but it's that's going to vary from isotope to isotope. But the number that determines what makes carbon is the number of protons only.
Right. So carbon 12, because 12 minus 6 is 6. That means carbon 12 will have 6 neutrons.
Correct.
Carbon 13 will have 7. Carbon 14 will have 8.
Yeah.
Okay, so the half-life is 5730, 5,730
Yes.
years.
Yes, plus or minus 30.
So, okay, that's fine. 5,700. So when it's formed, it has 11,000 change. Correct?
11. I'm not sure what you mean by that.
Let's pick a number, like pick a unit, like 1 gram or something, or kilogram.
Okay, a carbon when it a C14 or 14C when it's first created or made. Okay, the shelf life is 11,000 years
No.
ish.
No. So
Then
nuclear decay.
why does it say half life? Can't you just add 2 half lives and say this is the full life?
No, because there's no way to predict exactly when a single atom is going to decay. So let's say we take a look at, say, 6 particular carbon atoms, right? We would expect after 57,000, I'm just rounding here, after 57,000 years, we would expect to have 3 left.
Why don't you say 100?
Well, okay, fine, 100. Let's say we're watching them somehow. We've got the greatest microscope in the world, and for some reason we have 57,000 years to run this experiment. We're doing crazy thought experiments. We're time lords in this one. I don't know. So we watch 100 of them. Now, over the course of that 57,000 years, about 50 are going to decay. But we have no way to know exactly which ones will decay or exactly when. One might decay right away. Another one might decay in 50 years. Another one might decay in 1,000 years. We have no way to predict.
It's literally
exactly
impossible.
Right.
And it's actually impossible.
This is because this is all down to basically quantum mechanics, which I don't think we really want to get into. But what you can say about quantum mechanics is you cannot exactly predict what's going to happen to any one particle in quantum physics. All you can say is what will happen to a group of them overall as a result of probability.
Anyway, back to the analogy with 100.
So basically, when a carbon atom or carbon-14 forms in the atmosphere, it could decay immediately. It could decay after 57,000 years. It actually has a 50% chance of being gone after 57,000 years. But after 2 half-lives, which is 114,000 years, it doesn't have a 100% chance of decaying. It has a 3/4 chance of decaying, because every 57,000 years, it has another 50% chance. Right. So if you made it to 57,000 years, the chance of that atom decaying in the next 57,000 is still only 50-50.
Correct.
So rather than it's not like you have only 2 halves. It's like whatever you have that. And right. So you have the 100, so you half it, now you have 50, but you don't, but you have to half that again. So now you have 25.
Okay.
Right. So I half that again.
I get all that. I get all that.
Okay.
When carbon-14 is deteriorating, half a life is 5,700 years.
Well, that's not half the life of the atom.
That's half life of the group of the of the entire amount you have, right?
Right. So if I start with 100 carbon atoms, 57,000 years later, I will have 50 of them. But 57,000 years after I have 50, so now we're 14,000 years?
Yeah, whatever it comes out to be.
I'll have 25. Now, after another 57,000 years, so now we're, coming up on 200,000 here, I will have, 20, so...
All right,
12,
I get it.
Every 5,730 years, you're going to have half whatever your amount is left.
Right. Now, eventually, the last one will eventually decay, but it's...
And that last one will have existed several half-lives, right? And why it does is
okay.
unknown. Well, I have a theory on that, which I'll talk to Dapper about one of these days. But
here is my question.
Okay, yeah.
And it's important to me.
Absolutely.
When we find that bone in the I can't find where I wrote it. Sarantary, whatever you called it. Sarantary.
Quaternary?
Whatever it was.
Yeah.
Quaternary. Okay. We find a bone. We're in the quaternary zone, call it whatever. We bring it back, and we also bring back a rock with it.
Okay.
And we want to test it. What sorry, I got to back up. What dating process are we using? Are we using uranium? Are we using potassium? We can't use carbon.
Not usually, no. It would have to be
Right.
very recent to use carbon.
Yeah, carbon's not
And it would
really
have to
good
still
for
be
anything like that.
biological.
Okay, let's stick with dinosaurs then,
Okay.
because it's easier, because then we can completely eliminate the carbon.
Okay.
Okay, we find a dinosaur bone. What dating process do we use? Do we use uranium to
All of the
lead?
above. You can use any of them.
So,
But in the
Okay,
rocks
so-
that surrounds it, right? You're
I think
dating the rock.
so
No,
here-
wait a minute.
Okay.
Before you answer,
Right.
let's just say Steve said all of them: potassium, argon, uranium to lead. Let's just say we're using those two. Okay. Now, here's my question: is how do we know
Mhm.
how much potassium, how much argon, or how much let's just stick with potassium argon.
Okay.
How much
That-
how much the parent potassium
Okay.
was there, and how much the daughter argon was there.
Absolutely.
Let me
That's a great question.
answer this question.
How do we know? Well, first of all, number one, do we know the exact amount, or are we on the presumption that it's 100% like fresh, right?
Mm-hmm.
Let's just start with that.
Let me answer. I know where you're going with it, and I'll save you a lot of time. The answer to your question is pretty simple. We don't know, and we don't assume. And the reason being is it's irrelevant. When you're using what's called isochron dating, the apparent to daughter ratio is always going to be some fixed ratio. What it was is it makes no difference. You don't have to know how much at what beginning you had as like carbon. Carbon is not isochron dating. This is a thing that Hovind tries to conflate all the time. We don't have to know what the original parent isotopes were. It's irrelevant in isochron dating. So if you look at it's called geochronography. Looking at these formulas, they're very complicated. They're called Taylor series. But if you look at them very specifically, you'll find that some of the stuff falls out. It means it just goes away. It makes no difference what the parent isotopes actually are. So when you're asking, do we know? No. Do we need to know? No. It makes no difference to the isochron.
I'll actually point out that actually in the specific case of uranium argon, we actually do have a good reason to assume 100%. And that's because when rock cools,
That's a little bit different because of volcanic activity,
Right.
yeah, and inclusions.
So for that one, for specifically uranium argon, we actually have a good reason because argon being a noble gas is actually excluded from the crystals that form as the rock cools. It all goes out, and we know this because we can check.
Yeah.
And so for that reason, we can assume essentially all of the argon is as a result of uranium decay, because argon can't form into crystals because it doesn't make chemical bonds, because it's.
So we know that it's actually called intrinsic and not exogenic.
Right. So for that one, we do.
I have a quick question. I do have a quick question. And I don't mean this in any kind of derogatory way whatsoever. When you say when we find like skeletons and bones, we're not finding the same bones that were there anymore, right?
Sometimes we are. There's a number of different ways that
Depending
essentially
on
fossilization.
age, right?
So it's actually fairly common for the hard calcium parts of bones to be original. Hi, Jackson.
It's all been fossilized, right? For the most part.
Actually, it's actually not too uncommon for the original bone to still be there. And what's happened is the spaces inside the bone, because bone isn't just a solid piece of
permineralized.
Yeah, so sometimes minerals will have seeped in and filled in those holes, but other times, many of the time, the actual calcium has actually been leached out and other minerals have replaced it. So there's actually some fossils, the original bone is there.
Right.
And it's filled in with minerals. Some fossils, the bone has been entirely replaced with other minerals. And so there is both occur, basically. It's actually not that uncommon because bone itself is basically a rock. The hard part of your bone is basically just a rock that was created by osteocytes. In the same way that like a seashell is basically a rock created by a mollusk.
And that's why when
I just know that's kind of a misnomer sometimes. It's that people think it's the literal perfect bone.
It depends, because like when Mary Schweitzer did her work on the osteocytes, those are original osteocytes from the osteoblast. So that she could actually tell. I think it was the mandibular bone, if I'm not mistaken. But in yeah. So she could actually tell that that T-Rex had actually had offspring, and the reason why she was able to do that because we can actually look at ostriches and do the same thing. So
She could tell it was gravid. It could have been her first reproductive event. We can't technically say it. We can say it mated successfully.
Yeah. We don't know if she actually, if it came to fruition, and if it wasn't a zygote that didn't make it. But we know that she mated and she was fertilized.
Yeah, we can say that because otherwise you don't get medullary bone.
Yeah.
Okay, so does science use the same dating method on rocks and fossils?
Okay, so generally speaking, you cannot directly test rocks that aren't igneous. So what we do
what about metamorphic?
So one of the problems with metamorphic rocks is that because of the heating events, the chemistry will change, and sometimes things will come out of basically this. It's essentially a solution, but it's solid, right? So there will be outgassing and things like that will occur that will mess up the radiometric dating if you try to use it on most igneous rocks. So as a rule, unless it was laid down in a volcano and was not subsequently metamorphosed, you cannot directly test it. But what you can do is, because we know that you cannot generally lay down rock underneath rock that's already on the surface, right? It has to come on top. Like if you drop a ball, it's not going to land 3 feet underground. It's going to land on the ground. So when a new layer of igneous rock is laid down, it's always laid down on whatever the ground is. So we can say whatever's below that has to be younger. And if we dig down and find another even lower igneous layer, we can say that whatever's above that has to be younger. And of course, we take into account things like there's folding and things like that, but that's something that geologists can recognize. So as a general rule of thumb, when you go down, you're going older, although you can't say at the outset how much older. But then when you find each of these igneous layers, you directly date that igneous layer, and then you use the date ranges between them to relatively date
and of course it gets more complicated once you start adding in folding and raising and lowering of layers, but that is something that's why geologists go to school for so long, is to be able to recognize those things when they've happened.
Yeah.
All right, so let's talk about igneous rocks.
Okay.
Steve brought up earlier, someone brought up earlier, Mount St. Helena erupted in 1980.
Mm-hmm.
The rocks were dated 2.8 million years old.
They were, and here's the interesting thing about that. So they used various techniques that are generally used for rocks that are over, I think it's like over 100 million years or so. And so one of the things about this is that when you actually put something into the test machine, what it is going to do is it's going to essentially pulverize it and then analyze the chemical spectra that's coming off as a result of it's very complicated in physics and chemistry, but basically you see what kind of light you can get from them after they're excited, and that will tell you what elements are in there and which isotopes. And so but the thing is, when you clear out the test chamber, there's no way to completely clear it out, and so you always get daughter element signatures, even if you were to just run the test empty. And what 2.8 million means in this case for those dating techniques is lower than we can tell, because 0 is within the error range for a 2.8 million year date.
Yeah, and creationists latch on to that, and they don't seem to recognize what those numbers mean.
Right. It's sort of like if I had a yardstick and it's just a yard, it has no like feet or inches or anything, and you ask me to measure how thick one of my hairs is, I can just tell you it's a lot less than a yard, but that's about all I can do. And it's the same thing if you want to measure rock that was laid down igneously yesterday, but you use a technique that's used to tell you within 10 million years how old it is, then 2.8 million is a correct answer. Yeah, it's essentially the same as 0.
And you also got to remember that when you're dealing with volcanic activity, you're dealing with argon 40 and potassium 39 and argon 39, and these gases that you're only going to be able to tell when's the last time they solidified.
Yeah, and only within that error bar.
Yeah.
So basically, in this case, 0 is within the error bar of the technique used.
So that rock may have been a long time, obviously much older, but it liquefied and then got reconstituted, right? And so
the dating tells you the time it cooled.
It tells you only after the last time it's been liquefied and then cooled off.
Right.
And also you have what's called inclusions that can give you some errors as well.
It's sort of like if I say it's 2.8 million years, give or take 5 million years, well, we know it can't be negative, and we know 0 is within that range. And that's the kind of answer you would expect to get if you used something that's going to give you a 10 million year range on something that was laid down yesterday. You expect some positive number with an error bar that's going to include 0.
Yes.
Okay. Lava flow. Let's see. Lava flows at Mount Nguruho however you say that, in New Zealand, erupted 1949, 1954, but the rocks dated
Well,
275,000
for exactly the
years
same
old.
reason we just said, though.
So there's that. Also, I've seen a number of. Now, I would have to actually go do research that we probably don't have time right now, but there has also been. Okay, sorry.
I'm sorry, I have to ask. So you're saying the lava is older than the rock when it cools?
No.
The lava is the rock when it cools.
Yeah,
but
you can't. Let's put it this way: you can. Okay, Steve Austin did this. He's a PhD, but he is also a very disingenuous young Earth creationist. He used these techniques, knowing full well that you cannot use those techniques on that particular type of system. It's a very dishonest thing to do, because we already know you can't use argon or potassium argon dating to date volcanic rocks for these very reasons. But they decided to do it anyways, right? Because it gives them, some numbers to throw up to younger creationists and say, oh, look, this is corresponding to what we believe, even though it's completely BS and completely dishonest.
And the reason why is because the rock heats up so much, it changes the rock. Right. So
and also the gases go out.
Lava.
Right?
Well, it is still magma. It's broken down so much before it becomes rock again that you're basically have to start again.
Okay, so I'm at a volcano, and this all ties back to young Earth, old Earth, evolution. Okay, that's why I'm asking. I go to a volcano, and the lava's coming down. I get a sample, I put it in a bucket with some cold water, it cools down, I put more water, cold water, becomes a rock. Are you saying that you can't test that rock?
You can.
10 years from now and say, sorry, don't know how old this rock is?
No, you can test it 10 years from now, and what you will find out is that you will get back a number with a error bar that includes whatever age it actually is, including zero if it's brand new. So if I tell you I am, or if I tell you that I have a human, this human is 3 years old, give or take 5 years. I could have a newborn, because 3 years, give or take 5, would include a newborn.
That's what an error bar is.
But my question is, what dating method are you using on the igneous rock once it's cooled?
Any of them will give you the same type of result. It's just that each one of them has
carbon.
Carbon
any of the ones that can be used for igneous rocks.
Yeah,
carbon dating is only used for biological material.
Right, you have to have C14 to have carbon dating. Carbon dating is basically a way of looking at a ratio of carbon 14 to other types of carbon.
Right.
Without
it only
biological,
works on
right? Without having that. When we're alive, we're constantly renewing our carbon 14. But when you die, you stop doing that. So we know that the carbon dating from 14, we're no longer producing it, and so that's why we can determine when something existed, because it was no longer producing carbon 14 after death.
Right. So we can't use carbon. Carbon is strictly for things that have died and were once alive.
Right.
Living thing. Okay.
Living things.
Yeah.
Correct.
But all the other ones,
organic,
we use on inorganic things, so rocks, right? And
so
it's always
dinosaur bone found in the the Triassic, as you said. What dating method would we use on that?
I mean, I would have double check the exact date ranges that you use for each.
No. I'm asking, would you use
you would
potassium, argon?
Well, you would date the rock, not the bone itself, from that far back.
Right.
Right. So we're looking at if we see these dinosaur bones in the Jurassic period, and we say there's rubidium or strontium or uranium, lead, we can look at different materials, elements in that strata to say, okay, what is the best form of radiological isochron data we can use to make a determination. I mean, they're all pretty similar, but because you don't find any weird variations. Even if you use all these things to date the Earth, you don't find one that says 4 billion years or 4.5 billion years and another says 2,000 years. You don't see that. You see a range, right? So even if the one of them is the least accurate, you're still going to be within a few 100 million years. They all have a similarity to it. There's a consilience, and I think that's
also. Oh, sorry.
I was just going to say that's a hard thing for even the Rates Project to overcome. I mean, even the most staunchest young Earth creationists have a very difficult time when it comes to the radiometrics, because there's certain things they just cannot resolve no matter what they do.
Why do you think I'm asking?
Can I maybe turn this around a little bit? If young Earth creationism was accurate, what would we expect to see via these dating methods instead?
Wouldn't we still expect to see at least one of them come up with 7,000 years for the Earth or something? When none of them do, not one even come close. I mean, you're talking orders of magnitude difference.
That was a really good question, Cheshire.
She's smarter than she looks,
Thanks.
even in the mustache.
Hey,
For a 62
You're
IQ,
a fake
sometimes she,
mustache and a hat.
you think
Can
we
you
should
ask it again?
You think we just keep her around for her
I
looks?
said if young Earth creationism is correct, if that is the correct methodology, or that's the world view, whatever you want to call it, that we're going to use, if that's where is going to be our starting point, what would we expect to see when using these dating methods? So if we know the dating methods work consistently because the science, like that, the chemistry makes sense and the geology makes sense, but what we're not agreeing on is the conclusion of young Earth creationism. So if young Earth creationism is correct, what should these dating methods be showing? What do we expect to see?
Can someone answer that?
Yeah, actually, it's funny, Scott, because this is one of the problems I had with Dr. Humphreys. Because Dr. Humphreys tries to answer this very question with me. And this is all communication through email, so no, it's nothing I have online. But he actually posits, along with like the rate project, that there has to be something called accelerated decay. Accelerated decay is this stopgap ad hoc thing that creationists will throw out there. But when you ask them, what's the evidence for accelerated decay? They literally say, because it could be no other way, there has to be this because the Bible's right. They have no scientific basis for it whatsoever. Is there any scientific evidence whatsoever for accelerated decay? No, none, zip. It is just.
Well, I think that's not quite the answer to the question. I though I agree with you, I don't think that quite answers the question. I think more what would be more address the question directly would be, would we see different ages in the rocks? Would some of the methods not work?
We would expect to see.
What would we expect to see?
We would expect these to have around, obviously, an error bar. Let's say, okay, the Earth is 6,000, 7,000 years old.
Yeah, what would be consistent in those measurements? We would expect them to come back maybe 10,000 years plus or minus 4,000 years or something, right? Because that would be the tolerance. That would be in the error bar.
Right, and the ones with the even bigger error bars would basically come back with like a few million, give or take.
Yeah, the further back you go, the greater the error bar is going to be, right? So if you're only looking at something that we would expect, we want something around 7,000, so we would expect our error bars to be somewhat small. We're not going to have an error bar of 1 million when we're getting back stuff of 200, 300 million years ago. We would expect something to be around 7,000, 8,000, 9,000, 10,000 with an error bar plus or minus 4 or 5, which is going to be put in the realm of what creationists are saying.
I mean, you do uranium lead, even on a brand new rock, you're going to get a few million years because that's the kind of error bar we're going to get just because of how long the half-life is.
Yeah, exactly. It's going to be a few million years, but those are also going to be used to determine billions of years of age, right? So you're looking at billions of years of age with only an error bar of a few million years is actually nominal.
So would that essentially mean that all rock should be dated within the same error bar?
Yeah, the 7,000 years, or we'll just say 7,000. I don't know if the younger creation is very little. So whatever you think that the age of the Earth is, that number should be within the error bar for all rocks if there aren't any miracles going on.
Yes.
Now, the problem is once we introduce a miracle into the process,
which is Gentry, what he did, he called them singularities.
Yeah, throws the whole thing out of complete whack.
He literally did that to save his theory.
Physics no longer applies.
Correct.
Right? The rules don't matter.
Robert Gentry did the exact same thing. He wrote a book called God's Little Miracle, I think was the title. But he posited these polonium halos and tried to say that because of these polonium halos, the Earth had been cool within a few weeks. He didn't actually come out and say, from most of my readings with him, that he didn't say, okay, the Earth is 7,000 years old. What he claimed was the Earth is probably 7,000 years old, lines, because the Earth had cooled very rapidly in a matter of weeks to produce these polonium halos. But when the science went against him, and all these experts show that he was just completely wrong, he just fabricated something and said, well, I'm going to call them singularities, which are one-time events where God intervened to make it so, which at that time, obviously, Scott, you would have to admit, took it out of the realm of science.
Yeah.
Okay, so you can say with certainty and you can choose the level of certainty.
Okay.
That if we went to the
Triassic?
Triassic
Mhm.
and found a bone, we wouldn't date the bone, we would date
The
a
rock
rock.
that found it. Yeah, because that's 250 million years ago, give or take, right?
And now,
igneous or metamorphic.
well, almost always just igneous. Most metamorphic rocks.
Why? Why?
Because so what a metamorphic rock is, it is an igneous rock that, as a result of things like pressure and heat, has been chemically altered. This chemical altering can result in things like daughter elements leaching out of the rock, or additional of what would be parent elements going into it. And as a result, it is no longer reliable because of those chemical processes. Because anytime a rock undergoes a chemical process, the chemistry changes. And what we need is consistent chemistry. So if we have a non-metamorphosed igneous rock, we can tell that because it has been solid without metamorphosing for this entire period, because otherwise it would just be metamorphic, the chemistry inside the rock has not substantially changed, which is also why we get samples from inside the rock, not the surface.
Yeah, think about it. If you had an igneous rock that has some kind of like completely hypothetical, but let's say you had like argon in it, and all of a sudden you put it under high temperature, high pressure, and it starts going a fundamental chemical change would be metamorphized. That outgassing or leaching of the argon might have the argon obviously go out of the rock, right? So you're not going to be able to really tell anything about it because you don't have the actual original amount of argon in it to even begin to date it properly, because it's not there any longer. It's been leached out or outgassed.
Basically, we know that our assumptions about ratios, even in the cases where we don't need to use known parent-daughter ratios, because in many cases we don't.
Yeah.
But even then, we still have to know that the ratio of change is aligned with the half-life. And in the case of metamorphic rocks, we cannot make that assumption. Exactly. It is almost certainly false. So because that assumption of and by the way, when I say assumption, this is a conclusion of physics that is now being used as an assumption in geology. This isn't something we assume out of the blue. When science makes assumptions, it's because other people have already done the work.
Yep.
Like if I want to calculate how long it's going to take for a ball to hit the floor after I drop it from 5 feet, I don't have to rederive gravity. I can just say, I assume gravity, I assume it operates at 9.8 meters per second.
But you could, and you should get the same results either way, right? That's the
so in the same way, we don't have to redo all of the measurements on radioactive decay. We've already done those, and we continue to do those. But now if I'm doing geology, I can just take the results from those experiments, and now it becomes my assumption. So it's not an assumption in the sense where we just pick a number out of the blue. This is a conclusion of other parts of science. And then
we're over 2 hours, so I know that Scott wanted to keep it down to 2. So how about we do one last question and go from there? Because I know you wanted to keep it down to 2 hours. You told me so.
Me?
Yeah.
I'll go as long as you want.
Did you? Because you asked me. Didn't you say how long?
Well, it might be a good idea to stop at 2 hours and come back for another time, and maybe we can actually bring in a geologist.
Yeah.
I'm perfectly willing to come back if biology is
we can do part 2. Yeah.
And same for me.
All right. Well, then I will save. I'll save this question that I wanted to ask for the next time.
No, well, I don't know. Is it geology related?
Yeah.
Okay.
I mean, I'd like to hear it.
Yeah, let's put it on this one. Then we'll come up with some other stuff for the next one.
It's a 2-parter. You can keep your answer brief on the first and then elaborate into the second. A 100-year sample of decay rate is inadequate when talking about millions of years.
Okay.
A 100-year sample of decay rates is inadequate when talking about millions of years. Agree or disagree?
Disagree.
Okay, so the last one is, we do not know for sure if the rate of decay was the same 1,000 years ago, let alone 10,000 years ago, or 1,000,000 years ago.
Well, they kind of already discussed why it would almost certainly have to be the same 1,000,000 years ago.
Yeah, why?
I mean, and
go ahead.
I'm not a chemistry or a physics person, but I've seen articles about how there have been other factors testing, like it,
the fine structure constants.
Would this change over time? Would this change as a result of electricity or heat or pressure or water, etc.? And the answer is no, not really.
Yeah.
Yeah.
So
here's the thing, Scott. In order, okay. Well, let me read the super chat real quick. Athens 619, $5. Here's a serious question to everyone. It just went off my screen. Are rocks atheists? No. No. But thank you for that super chat. So let me discuss this for a second. If it was not the case, if it was, as Cheshire points out, and we always keep on saying on Caffeine Corner, what would we expect to see? If it was the case that the decay rates can change. Now, there are sometimes they can change, right? There are very specific times and in very specific ways. For example, what's called K capture, which can have about a 0.8% variation in certain types of isotopes. It's only going to be happening in isotopes. We have a very specific type of suborbital, which in the S shell is going to allow for electron capture. Okay, so it's not going to be on like all isotopes. And you also going to have metastable isotopes like beryllium-7 star that can actually have a very fundamental difference in their decay. But these are very, very specific things. But the actual changing of a half-life would require a fundamental change in natural constants. And if you change one natural constant, you change them all in some ways. Something else has to change, which means the speed of light's going to change, which means that the gravitational constant's going to change, which means that something like what's called the fine structure constant, which is Summersby constant, it's basically 1/137 if you want to like round it. But that number is a very, very important number. With that number changes, you change the Coulomb barrier, you change the repulsion of different types of the protons are going to be repulsed at a different
Steve, I think we can sum it up with you have to break physics.
It's bad. You literally break physics.
Can I
all of physics.
Actually touch this a little bit? I kind of want to get my little goblin hands around this. Yes, I'm going to
read the super chat. It's all yours, I promise.
Think that, okay, Res for 50 said, one in chat if you want more young Earth creational content.
And we want Res in our next chat. So
if you want a lot of that,
correctional content. There we go. I did it.
Yes, there we go. Yay.
My channel and Jackson's channel, if you want a lot of that, because I mean, I agree we should have more on the channel, but if you haven't seen either of our channels, we have a whole bunch of that content.
You guys are, and by the way, their links are in my video description.
And, yeah. So if you, I think I'm going to answer this maybe a little bit more simply, and it's that these things are the way they are, and there's no reason to think that they have or would have changed in the past or in the future outside some kind of massive event or some kind of divine act or miracle.
A massive event wouldn't be sufficient. It would have to be a miracle.
So right. So the only way that it's possible that these rates would have changed is by some kind of divine intervention. Yeah. So if you want to posit that there was divine intervention and therefore that's why the Earth may be younger or older than what people say, which I am curious as to why there aren't like older Earth creationists. Like you've got young creationists, you've got the ones that agree, but there are?
Yes.
I've never met one.
I work with Fuzz Rana is an old Earth creationist.
No, she means why aren't there people who believe that the Earth is like 1 trillion years old?
Oh, I got you.
Yeah.
Oh, okay.
There are like sects of Hinduism that believe that, if I remember correctly.
Yeah.
There are groups of Hindus that do believe that.
So let me point this out because somebody pointed this out.
Either way, point being is that if you want to posit that there was some kind of divine intervention, you would need some kind of justification for that.
Yeah.
And I don't think there is any strong justification for that. Because that would just basically be like, well, God's tricking us, and it doesn't. I don't recall off the top of my head anything in the Bible that would indicate.
It's kind of like the old phallus hypothesis. It would be last two Thursdayism kind of thing. But
I mean, maybe you could make an argument for it based on what the Bible says, but I think that you would probably find a lot of contention trying to make that argument. There's also a super chat from Berillion for 5 that says, does Williemaysfan apply the same questions to the proposals of young Earth creationists as he is asking here?
Before we get to that question, I want before it goes too far off the screen, Don Giovanni brought up a very good thing. He talks about the Oklo reactor. Scott, when you get a chance, look up the Oklo reactor. The Oklo reactor is actually a very well-known natural. There's a few natural reactors, but this is the very few ones that's been self-sustaining itself for over a billion and a half years. It's about 1.7 billion years. So it's spelled
OKLO.
Yes.
Reactor.
And what that tells us, because that reactor is a natural reactor that actually reached criticality 1.5 billion years ago, that the fine structure constant has not changed in at least 1.5 billion years. There's no possible way that has changed in 1.5 billion years. So when you're saying that, hey, these decay rates have changed, we can definitely prove by using physics, and I use the word prove here, obviously in the science sense, but we can
result in very high confidence.
Yeah, prove it in a very high confidence that we know that the fine structure has not changed, therefore the decay rates have not changed. Because if it had changed, the decay rates have changed, then that fine structure would have changed, and of course we don't see that, and so we can't verify these things.
Mm-hmm. And also, I would point out that when a process relies on fundamental aspects of physics, very short observations are valid to extrapolate to very long periods of time.
Yeah.
So, for instance, when we know that the speed of light is always constant for any given observer. So, as a result, we can say how long would it take light to go any distance. But because since the speed is always the same, you just use the speed triangle you probably learned in like 7th grade or something. You say, okay, we know the speed, we know the distance, what's the time? Or we know the time, we know the speed, what's the distance? And so you can observe the speed of light for 30 seconds and make that observation valid for 10 billion years.
That's also how we know the lowest bound condition for the age of the universe is 167,000 years old based upon supernova 1987A. For that very reason. Okay, so Virillion asked that question. Does Williemaysfan apply the same questions to the proposals of young Earth creationists that he's asking here? Do you ask them the same question, Scott?
Yeah, I understand the question. Young Earth creationists don't have that conversation.
That's interesting.
What? I'm sorry, I'm a little bit confused. They don't have the conversation. There's no discussion amongst young Earth creation about whether or not these dating methods are valid.
Oh, that's what the question was?
Well, I mean, that's
they
one
were
of the,
asking if you.
Yeah, I mean, that's one possible.
Well,
question.
for instance, that you're asking us today.
I can tell you right now that they do.
Ask...
Look, they actually do. Young Earth creationists actually have all, I mean, the experts, right? The people that have real degrees in the Young Earth creationist community have all said, Scott, these radiological dating methods do work. That is their words. They agree that they do work. They've changed their position decades ago. The RAPE project was an abysmal failure, and they did have to concede that these dating methods do work. However, then again, they start positing other things like accelerated decay, and they start trying to do these ad hoc explanations,
sure,
right? Because they can't admit that the results are correct.
I think.
Another example in within evolutionary biology, they try to figure out like what organisms are related to other organisms. How far can we take common ancestry back before we start running into problems? And the answer is immediately. Yeah. But they try to posit a sort of rapid speciation. Go ahead, Jess.
There's I think I might be able to rephrase Brilliant's question a little bit easier. So just so it might, do you with the same, are you consistent in your scrutiny that you're giving us here with young Earth creationism?
Yes, I like that phrase.
I'm not scrutinizing any of this.
What do you mean?
Okay.
It seems like you're asking very good questions.
These are all honest.
Being scrutinized. Yeah, no, that's not a dishonest thing.
We're honestly asking, sure.
Yeah, none of this is dishonest. It's nobody's suggesting that. It's about.
We are pro scrutiny.
Yeah, exactly. Yeah.
Well, scrutiny might be the wrong word, but I think what that person's trying to ask is, do I speak to other young Earth creationists and ask similar questions to see if theirs or our responses are the same, or if we make up excuses? Or I can tell you, for me personally, when I talk to Christians, we don't talk about the age of the Earth. We don't.
Then how did you come across young Earth creationism then?
Kent Hovind.
There we go.
I hope you are starting to realize that Kent Hovind is a con man. You do realize this, whether you agree with some of the things he says is completely tangential. He is a straight up con man.
But since I first heard it from him, I do watch a lot of videos. Dr. John MacArthur, James White, probably throw out 3 or 4 other names that you might not know or probably don't know. Sam Shmoon. Anyways,
if you
might
could
be
actually
surprised
give me
I've
a
heard of
list
every one of
of
those.
Yeah, if you don't mind
Yeah, I
giving
know- I
us
know,
a
like-
list, because we'd be happy to look into them and maybe have conversations with them also.
Yeah, I know who they are.
I think it's fair to say that, well, a friend of mine, RJ Downard, and I, we wrote a whole book in which we critique creationist arguments relating to biology, geology, paleontology. When you look at the arguments of one creationist, of one professional young earth creationist, when you see the tactics that they employ
Oh God,
to make their arguments,
so bad.
you realize, you see it in one and you think, okay, well, surely they're not all like this. So you look at another and you look at another and you start to realize they're all the same.
Yeah.
Yeah, I
They're
mean,
all using the same. It's really sad actually, because Georgia Purdom and Bodie Hodge used the crocoduck argument
oh God.
that Ray Comfort uses.
And she has a real legit degree.
She has a PhD in genetics, and you think she would never make this argument.
It's
She's not
bizarre.
You know, she is not dumb by any means, and she knows better. And yet she made this argument. They published this in the New Answers Book 1.
Do you think
Now-
she's just straight up being dishonest for whatever reason?
I
don't know what other reason could be.
Here's
I'm
what
not
I'm
going to
gonna
speculate on her psychology, but at the same time, though, when you see all of them using these same tactics, you have to realize something's wrong here.
Well, I'll give you
Something's wrong
a specific
entirely.
example, like for Tompkins' work, right? So Tompkins was basically trying to criticize the human chromosome number 2 fusion site, and
Right.
he tried to argue that there's a defunct pseudogene called DDX11L2, and he made the claim. This was his words. He made the claim that there is a functional transcript that comes from there, from the canonical site. And when Ruth had actually inquired about it, and he said, okay, where are you getting this from? Because it's getting a 3.7 RPKM read from the testes, but then other than that, there's just really nothing from it. But he's saying that it's highly expressed, Jackson. And so when we say, okay,
Right.
what data are you getting this from that you have a highly expressed transcript from this gene? He has no data to show anybody. He just accepts. He just says, creationists have to accept that's what it is, rather than, okay, well,
Yeah, when
we're
you-
not a creationist. We don't have to accept your word. Where are you getting this data from that is highly expressed? And he's never
Right.
been able to produce it.
Yeah, exactly. It's the same. It's the same sort of shtick across all creationism. Whether you look at Georgia Purdom or Andrew Snelling, who again another PhD, he's actually a geologist, and he was using these articles on these researchers who made abiogenic oil in a laboratory to say, well, therefore, biogenic oil can form in a couple of years
Okay.
in nature. And of course,
Those are not
that's
the
not
same
at
thing.
all what the papers were saying.
Yeah.
Right.
Oh, by the way, no,
and yet-
I forget. Tompkins actually did try to pull a fast one. He tried to use expression levels for DDX11 is a family, right? So he tried to lose expression levels from DDX family, and then say, "Oh, look, it's for the DDXL2." And Ruth called him out on
I
that
do
too, I
have
believe.
one
Okay.
question, since
Yeah.
we do need to start wrapping up.
Yep.
And maybe this question can be answered next time, but it's essentially, how much time do young Earth creationists tend to spend speaking to other religious people of the same religion who don't think the Earth is young? Because I know young Earth creation is actually pretty new comparatively.
Well,
The modern form certainly is.
Yeah.
the circles that
Yes.
the circles that I run with, the people that I associate with, are all Bible believing people. We don't. I can only say from my experience. I don't even watch Dr. Hoven anymore.
Sorry, we got to laugh when you say that.
I approve.
Yeah.
I don't
Gee, I
watch him
wonder why.
anymore.
Yeah.
I don't watch him anymore because I'm more of a Psi 10 Bruggencake guy now.
Oh, yeah,
Oh, no.
Well, I'll tell you what. I'll get Cy on if you want. You want to hang out with him, and you guys could tag team us if you want. Because I mean, I destroy Cy, and he knows it. And
That would
but I
be
like
a
Cy.
lot of fun.
I mean, Cy is a lot of fun. I gotta admit.
I don't know if I mean he's like all philosophy, and I don't do
Yeah.
philosophy.
So
Well,
I
that's
think
why you would need a little bit of column A, a little bit of column B. You get one philosophy
How about this?
person, one
How about this? How about this, Jess?
biologist, one.
How about this,
you know
Jess?
what I mean?
How about this, Jess? Scott here and Cy 10 against me and Ozzy.
Well, Sai knows me.
Yeah, yeah.
oh, that's
We've spoken off. We've spoken on Skype a couple of different times. So it's, if you said my name, Willie, like he would know me immediately.
Yeah, I'll wait. Well, Resident.
Scott, there are a lot of Christian content creators on YouTube who accept evolution, like Inspiring Philosophy, Michael Jones, Sentinel Apologetics, and Cygar. Yeah, there are loads and loads of
Capturing
Christians on YouTube.
Yeah, I remember one time I called Cygart a fake Christian, and the whole chat went at me.
Yeah, they will, because
Yeah, that's kind of not cool.
But here's the thing,
Yeah.
Resident, since as he said, if you want more, YEC content, we are going to do stuff like this more. I'm going to bring in people. I don't know how much formal we're going to have. I mean, I kind of like this whole just have a hangout and just talk. I don't think it needs to be made into any kind of like real production value, but I miss those things. I miss these kinds of discussions. This is how my channel started, doing these types of discussions.
Mm-hmm.
Steve, I have a lot more questions, so.
Well, see, the only problem is that we've gone for 2.5 hours, and people tend to tune out.
Yeah,
after
that'll be. For
No,
next time.
we can do it again. That's
yeah,
fine,
okay. So yeah, perfect.
yeah. And
so-
I'm perfectly down if you want me here to come back and answer
Who
more
are you
questions.
again?
Same for me.
Who are you
Yeah.
again? I'm sorry.
I'm Dapper Dino.
Oh, okay.
You might
sorry.
remember me from a science stream you did about nuclear physics a while back.
I do remember that. Yeah, I've done a few nuclear stuff. All right, so let's wrap it up, guys. Scott, like I said, I do appreciate the fact that you come in with an open mind and willing to ask some questions. Although people have said that you've probably heard a lot of these answers before, but we take the opportunity to talk because we've had somebody like Wayne Fillmore, who was a young creationist. He eventually realized it was wrong, and he did change his point of view. It does happen. It is rare. We've had a few flat earthers change their position over the years too. We see it, and again, we may not have a hell of a lot of people changing their minds, but at least this is out there. So if there are people that do want questions, they at least have the resources to know who to ask, right? If they really want to know a lot about this, they go to Dapper's or Jackson's channel, and they'll just ask them. I mean, they're very accessible. They're not superstars yet. Give them
I'll say this lastly, and thank you for having me on, and thank you, Cheshire and Dapper and Jackson and everyone watching. I'm really my worldview is slightly beginning to crumble when you brought up the speciation and having that be macro, because I believe in speciation. And
that's what got. By the way, that's what got Wayne Fillmore too. He actually.
A lot of us have been there.
Yeah, he
a
actually
lot of us have been there.
said one of the reasons why he started to start leaving Earth Creation and look into it more because he felt he was lied to. Because I did a debate with him on macroevolution, and I explained to him speciation, I explained to him allopatric and sympatric, and he was like, wait a minute here. That's actually in biology what's considered to be constitutive of macroevolution, and everybody's like, yeah, what do you think it was? He's like, well, that's not what I hear from young Earth creationists. I'm like, well, they're lying to you, and he found out that was the case, and that's objective. I mean, this is not just us going, oh well, believe us over them. It is you could go look for yourself, just like he did.
Yeah,
that certainly also doesn't mean that all young Earth creationists are lying.
No. I mean like the
just to be clear.
I literally mean like the Thompsons of the world and the Austins of the world and Perdome. I mean, I don't know Perdome. Yeah, I don't know Perdome. I think Pelagia knows her, but I don't know Perdome. But I do know the other ones. But I will tell you this. Well, let me put this on record. I will tell you this. It's critical to know that even though these young creationists, PhDs, a lot of them do actually very much believe it. I know Humphreys, for example, very much believes what he puts out. It's not an act with him. This is what I'm saying, that it really depends on who you have to talk with, because Perdome, I have some severe questions with whether she believes this nonsense or not. But like Humphreys, I've had numerous conversations with Humphreys and with other experts in that field that he really does believe his stuff. But even he has come around a little bit. Even he finally recognized that he had a mathematical error in something that he wrote in the journal Creation. I mean, so when they start recognizing things, what else can you ask for, right? That's what we try to do, is say, hey, look, maybe you're not quite correct on this.
Yeah.
By the way, you guys want
Get
to
the
hear
hell
my,
out of here.
well, I'll leave you guys with this if Dapper's interested. It'll take like 1 minute. Do you want to hear my kind of hypothesis on causation?
No. Save it for next time. If we're gonna listen to
All right,
anything,
we'll do the next time. So
listen to
tune in next
Dapper.
time, and I'll give you guys.
Steve.
Hang on.
The last thing. Listen.
What?
The last thing we should do is have Dapper read Old English again.
No, it was hard on my ears. but I will give you guys a hypothesis of how, when we talk about, you know, if there's a causation for a decay event, I'm trying to formulate what could possibly be again, just bizarre, what we consider to be a possible causation. How about that? It'd be interesting. All right, go ahead, Dapper. Read us your old English.
Some more old English. I closed out the window I had that had the prodigal son, so I'll just read something random from Beowulf. Let's see, what's a good place? Sonathat Yawason Snotra Keorlas, but so that's going into the modern English section. Sorry. Da them mid Hrothgare on Holmul Liton that was youth ye blonde el ye menged brimblodfach blonden fjage gomele unbegomde ongeador sprakon that he that's Athelingus Efnewenden, and there's a break in the clause there, so I'll just stop.
Yay!
Someone check him.